Macha, ready to rock. Hello, my dear children. Jiggy, 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 jiggy. Let me see if at all I am audible and visible. Yes. Hello, dear children. So, yes, no wastage of time. I hope you know this is important. I'm going to directly take everything from your question bank. Okay, so make sure you send this video link to all of your friends such that everyone can get great marks in their MCQ section. Good morning. Good morning. Okay, so your ma'am have already cracked up with lots of live things like I'm really great. Thank you so much for asking. Okay, and as I said, make sure you share this video link with all of your friends who are almost done with biology. You have to be very sure about MCQs because we are going to do that. And everyone who has not yet hit the like button, click on the like button and subscribe to the channel. Let's go. So listen to me carefully. I am here with one very important thing, that is logic. Okay, so if at all you see the blueprint, we can clearly see that there are some chapters which give more MCQs than the other. For example, human reproduction gives 2 questions, there is evolution given 2 questions, human health and diseases giving 3 questions. So what are we going to do is that as much as we can, we are going to complete the MCQs from these chapters which have more weightage. I'll be taking the live only until 9:30. So until 9:30, whatever chapters are being done, they will be done. These will be the high weightage chapters out there according to MCQs. Okay, so let's start with the first chapter that we have, and that is human health and diseases. And are you ready? Everyone, super ready? Yes? Will MCQs come from here? I'm not sure whether it comes from here or not, but definitely we can try, right? Trying is also an option. Not referring anything and trying is definitely an option, right? Perfect. Okay, so let's begin with the first question that we have over here. We have to be a bit fast, okay? And please do the question according to our pace. We'll go together. The first question says, which of the following pairs contains an infectious and also a non-infectious disease respectively? So you have to tell what is infectious, what is non-infectious. First one should be infectious over here. So one will be infectious, typhoid and AIDS, AIDS and cancer, pneumonia and malaria, cancer and malaria. What do you think is your answer for this particular question? Ma'am, what about the diagrams, dear? I had made a separate video on it. Please go and watch it. A, Kid Zone says A is the answer. Think, guys, don't get it wrong. First B, okay, Manish, Kirtana and Yash and Manish T also, Ishant also says B. Guys, B is the answer. Listen, AIDS is something that is infectious, sexually transmitted. Cancer, do you think it is infectious? Someone who is having cancer, if they are beside us or probably there is anything out there, do we get it? No, it is non-infectious disease. So answer for this question is going to be B. Okay, clear? Perfect. Next question. Which of the following is not a bacterial disease in human beings? Which of them is not a bacterial disease in human beings? Pneumonia, malaria, plague, diphtheria. Which one is not a bacterial disease? Think of all the bacterial disease. You have typhoid, which is bacterial disease. You have diphtheria. You have plague. You have dysentery, plain dysentery. And you have pneumonia, which is bacterial disease. You have been asked which one is not bacterial over here. Okay. My God, awesome. Everyone is here. Tanmaya, hi. Srikesh, hello. Jia. By the way, if at all I'm missing out any names, I'm literally sorry. Okay, 2B. Guys, can you give me the question number also? 2A, whatever. 2, you put 2 number. Okay, question number is important. Otherwise, I will not understand which question you're solving. So, most of you guys are saying it is B, and B is what? Malaria. Yes, malaria is not a bacterial disease. It's a protozoan disease, right? Yes. Going ahead with the next question. I hope you guys are understanding. I'm going fast. Why? Because you do not have time and we need to study a lot of things. Perfect. Now, send this video to everyone out there. Let's make it 100, more than 100. Let's make sure we get so much marks in MCQs. One of the following is not the causal organism for ringworm. One of them is not going to cause ringworm. What is that? Microsporum, trichophyton, epidermophyton, macrosporum. Over here you have 4 options. This is question number 3. Answer like that. 3D. Sanjeevani says 3D is the answer. Zainab says D. Jaya says D. Mansi D. Mansi has a different answer. Okay. People, entertainer, Numan. Okay. Ishant. So I see some names and over here macrosporum is being mentioned. My dear children, Ringworm, I told you. Trichophyton, epidermophyton, they are fighting. Fighting for what? Small spore. What do you call the small spore as? Microsporum, not macrosporum. So this one is odd one out. This one is wrong option. Okay. Going ahead with the next question out there. And it goes like this. For that, I have to go up. Okay. So let's go. Intestinal parasite which causes blockage of the intestinal passage and whose eggs are excreted along with the fecus of the infected person. It's an intestinal parasite, please make sure, and it is causing blockage of the intestine. Only one guy can do it. Think about it. This is question number 4. Question number 4. Come on. Fourth answer is, please guys, write the answer like 4A, B, C, D. That will be really good. Okay, 4A. People Entertainer 4A. Mansi says 4B. Okay. Manish says 4B. Rizwana says 4B. Albert. Okay, Albert. Don't go ahead, please. Okay. Yash. B. Okay, fine. What is B? Wuchereria bancrofti. B is ascaris. Ascariasis is the disease where intestinal blockage is there, right? You have intestinal blockage, you have anemia, you have internal bleeding, all of this, please remember, it is ascaris. Fifth question, elephantiasis, a chronic inflammation that results in gross deformities, is caused by. Listen, it's a chronic inflammation. Inflammation is happening. That is why elephantiasis. Okay, it leads to gross deformities. What is it, the causative agent? Ascaris, E. coli, Wuchereria, or Trichophyton? Fifth, C. Okay, Aditi, Ayu, Joshitas, Jaya, Ishant, XMRIT, and Zainab, everyone, Kid Zone. My dear children, I'm literally happy you're giving me the answers. I'm so sorry if at all I'm not able to read out your names. It might save time, that's the reason. Okay, so 5C is what I'm mostly seeing. C is what? Wuchereria. Yes, Wuchereria, what does it do? It affects our lymphatic system. It affects our lymphatic system. So what happens? Usually lymphatic system is the one that collects all the fluids out there, the extra fluid, such that there is nothing left behind. Okay, and if at all, you only think, if at all fluid is there, it will lead to inflammation. Right? So lymphatic vessels is the one that is being affected. Okay, next question. Which of the following is affected by the infection of Wuchereria bancrofti? The same thing that I told you right now, and you know the answer for it. Come on, guys. Perfect. It's your army who are going to rock tomorrow. Yes, we'll rock tomorrow. Perfect. I didn't know that I had an army. It's so lovely of you. Okay. Namaste, dear. Okay. Yes, 6th A is the answer, and that is lymphatic vessels. Always remember, Wuchereria bancrofti, elephantiasis, filariasis, all the same thing, and basically it affects your lymphatic vessels. Going ahead with the next set of questions out there. Which of the following disease is transmitted by bite of female Anopheles mosquito vector? Anopheles mosquito is not given. Female mosquito vector. So which of the following disease transmitted by bite of female mosquito vector? You have options like what? Filariasis, amoebiasis, typhoid, pneumonia. What do you think is the answer? Ma'am, can you please help me in Paka questions? I want to... Okay, that went. Dear child, if at all you join the WhatsApp channel, there is a heartfelt message that I have written out there. Okay, please make sure you go through that message. I have given the message especially for the children who probably are lagging behind at this particular point of time. Go through that, okay? Yes. 7A, 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 7C, 7C. 7A and C is what you're confused with, is it? A and C. Typhoid. Mosquito vector you're talking about. There are only 3 diseases. No. Basically 4 diseases that are caused by mosquito vector. Everyone hear out carefully. First one, malaria. We know it's female Anopheles mosquito. Next 2 things is basically dengue and chikungunya. Dengue and chikungunya, both are viral diseases. They are being transmitted by the bite of Aedes mosquito. Aedes mosquito. Now, in your NCERT, one very important thing is written, that is Wuchereria bancrofti causes filariasis or elephantiasis. But who transfers it? It is again by a mosquito's bite, and the name of the mosquito is Culex mosquito. What do you call it? Culex mosquito, and most of us miss it. So the answer for this question is what? Filariasis. Okay. Why is it not working? Okay. Come on. Yeah. The answer for this is filariasis. Perfect. 8. Okay. Now 8th question. Which of the following pairs correctly matches a disease and a pathogen causing it? 8A, 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 8A. 8A is what? 8A is what? Typhoid, salmonella typhi. Does it match together? Yes. Typhoid is actually caused by a bacteria, salmonella typhi. Pneumonia is caused by Haemophilus pneumoniae. No, Haemophilus influenzae. So this part is wrong. Malaria is caused by Plasmodium. Ringworm is caused by Trichophyton, Epidermophyton. And what? Microsporum. What are they fighting for? The Microsporum. Cool? Yes. Now, going ahead, we have 9th. Appearance of dry scaly lesions with itching on various parts of the body are the symptoms of. You have itching, you have dry scaly lesions. Where do you see that? Elephantiasis, ringworm, Ascariasis, amoebiasis. What do you think is the answer for this particular question? Please, 7th. Just a second, okay? 7th question is what? Dear, I told you mosquito vector, even though we say that Wuchereria bancrofti causes the disease, but it is transmitted by a mosquito vector. A bite of the mosquito is the one that will be transferring the Wuchereria into our body. And that mosquito is Culex mosquito. The name of the mosquito is not given, but it is written in your NCERT that it is passed by a mosquito. Okay? Okay. Perfect. 9B. 9B, appearance, okay. So appearance of scaly lesions with itching. Whenever the term itching comes, just understand it is ringworm disease. Ringworm is not a worm, basically. It forms a ring-shaped structure which looks like a worm. It's a fungal disease, okay? Yes. Now, hemozoin is dash, precursor of hemoglobin, toxin released from streptococcus infected cell, toxin released from plasmodium infected cell, toxin released from Haemophilus infected cell. What do you think is the answer for this question? Sanjeevani says 10C. People entertainer 10C, and yes, Rizwana 10C. Some people are saying fast. My God, if I go faster than this, probably I'll die here itself. Okay, so basically the answer for this question is going to be C. Why, Baba? Hemozoin is what? Hemozoin is being formed in the RBC. In the RBC there is hemoglobin, right? Right. Now listen to me carefully. The plasmodium which enters, it breaks, it wants the globin only, not the heme. So what does it do? And heme is toxic for it. So it converts the heme to hemozoin. It converts it to hemozoin. Now when the RBC bursts open and hemozoin is released, that's when we get fevers and chills. What does that mean? That hemozoin is toxic for us. Heme is toxic for them and hemozoin is toxic for us. So toxin released from plasmodium infected cell, that is the answer 10. Done? 10th answer is C. Perfect. Awesome, my dear children. You guys are doing super great. Perfect. Next thing. A toxin substance responsible for chills and high fever reoccurring every 3 to 4 days in malarial fever is what? Interferon, hemozoin, hirudin, cholesterol. Check, colostrum. What do you think is the answer for this particular question? When this class does end, it ends at sharp 9:30. Okay? B, 11B, Finaz, Rizwan, I'm seeing multiple names are there. Okay, so toxic substances responsible for fever and chills every 3 to 4 hours is basically hemozoin. I told you hemozoin, for those of you who were not able to understand about hemozoin, listen to me carefully. Please give me 1 minute gap, okay? It's basically plasmodium is there, it is entering our RBC, it is feeding on hemoglobin. It does not want heme, it just wants globin. What does it do with the heme? It converts into hemozoin. Okay, it converts into hemozoin. Now, hemozoin, when the RBC burst open, the hemozoin is going to be released, and that's when we are going to get fevers and the chills. Okay, guys, we have to go ahead. During the life cycle of plasmodium, gametocytes develop in. Where does the gametocytes develop in? 11B, 12th A, 12th B. Okay, A and B is your option. During the life cycle of plasmodia, gametocytes develop in. Always remember, there are 2 things only that you have to remember: sporozoites and gametocytes. The way it enters our body, sporozoites. It enters through sporozoitic stage. It leaves our body in gametocytic stage. Okay, the last cell that the plasmodium affects is your RBC. So in the RBC, the gametocytes are going to be formed. They are going to be formed. Always remember. So your answer for this question is going to be human RBC. No sexual reproduction happens in the human body. It happens in the body of the mosquito. So the mosquito, suppose I have malaria and the mosquito comes and drinks my blood. It takes in which stage? It takes in gametocyte stage. Okay, now when it comes and basically bites kid zone, what is it transferring? Which stage? It's sporozoid stage, which is getting transferred. Always remember that. Now, 13th question. Which of the following is a viral disease? Pneumonia, typhoid, common cold, malaria. Pneumonia, typhoid, common cold, malaria. There are almost 40 questions, I think, from this chapter. 13B, 13B, 13C, 13B. Okay. Which of them is a viral disease has been asked? Pneumonia, typhoid, common cold and malaria. The answer for this question is going to be C, na baba. Common cold is the disease which is viral and it is caused by what? Rhinoviruses. It is caused by rhinoviruses, macha. So your answer for this question is going to be this. Pneumonia, typhoid is basically bacterial and this is protozoan. Yeah? Yes? Clear? By the way, everyone, is all going fine? Is it fine? Please give me a thumbs up if everything is fine. Spurti here, ma'am. Are you okay? I'm good, dear. Thanks for asking, though. Okay, next question that we have over here is, human immunodeficiency virus primarily attacks and destroys what? Human immunodeficiency virus, HIV viruses, primarily attacks and destroys. What does it attack and what does it destroy? Any idea? Any idea? Thank you guys for giving the thumbs up. Primarily attacks. So always remember, it wants to make sure that our immunity is low. That is why it needs something. 14th B, it is T lymphocytes, especially what lymphocytes? T helper lymphocytes. What is the HIV factory? It is macrophages where these cells are going to divide. HIV is going to divide. But the actual target is T helper cells, do not forget that. Okay. Now, which of the following is an autoimmune disease? What do you mean by autoimmune disease? Your cells are not able to recognize what is yours and what is not and basically kills up everything. What is the autoimmune disease? Rheumatoid arthritis, tuberculosis, malaria or AIDS? I am seeing Suresh saying the answer is A, Kirtana says A, Rahul says I'm stressed. Or, dear, listen to me. If you feel that you have a lot to study, please go out of this live. You study there. I do not mind it. Okay, listen, this video is not for views. This is for the children who feels that, okay, I just want to go ahead with MCQs now. Okay, so please take that into consideration. Do not be worried if at all you're not able to answer over here. I agree that you might have a lot of things to do. Apart from that, please do that. No problem. 15th A, 15th A, 15th A. Your answer is what? Rheumatoid arthritis. This is going to be an autoimmune disease. Okay? Next one. The chemical secreted by virus-infected cells that can protect other cells from further infection. So suppose a cell is infected by virus. What does it release such that the other cells are protected? Such that the other cells are protected. What do you call these cells as? Secretions as? Serotonin, colostrum, interferon or histamine? What do you think is the answer? C, 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 C. Ma'am, you're so beautiful. Thank you so much, dear. You're also beautiful. C, C, C. Okay. Perfect, are you? No problem, dear. Okay, so 16C is what? 16C is interferon. Okay, it is going to be interferon. Perfect. Interferons are the ones that are released by the virus-infected cells. Next thing, antibodies present in colostrum which protect the newborn from certain infection is, suppose a newborn is being born. It does not have antibodies for itself. Mother gives the antibodies. What is the name of that particular antibody? Any idea? Any idea? 16C is the answer. 16C is the answer. Yes. 17th question now, guys. 17th A. Prajwal, hello dear. 17th B. Oh, 17th B. Joshita. Okay. Sanjana says 17th. Oh, just a second. Sanjeevani says 17B. Okay, I have A and B options out there. The answer is A. I'll tell you a way to remember it. Mother. Sometimes some people call mother as Amma, right? Mother as Amma. So IGA, A for Amma. Always remember that. Okay, from Amma, the milk that goes carries IgA. Apart from that, when the mother and the baby is there and the baby is inside the womb, there also IgG is transferred. IgG. And how do you remember it? Always remember it. Gamed. These are the antibodies out there. G comes first and that is transferred through placenta. During allergy, it is IgE, allergy, IgE, all those things. Okay? Next, which of the following is not a lymphoid tissue? Spleen, tonsils, pancreas, thymus. Which one is not a lymphoid tissue? Guys, you're going to get 3 MCQs from this chapter. Do not take it lightly. Which of the following is not a lymphoid tissue? Spleen, tonsils, pancreas, thymus. What is your answer? 18C. Sanjana Sanju says 18C. Kirtana says 18C. Okay. Dawood, I have explained everything before that. That's why I'm not. Your explanation was too good. Thank you so much, dear. Okay. 18C, 18C, 18C. Okay, C is what? C is pancreas. Yes, it is not a lymphoid tissue, right, macha? Spleen, tonsils, thymus, all of them are lymphoid tissue, but not this guy, that is pancreas. Odd one out. Okay. Now, going ahead, what do we have? The site where lymphocytes interact with antigens and proliferate to become effector cells. The site where lymphocytes proliferate to become effector cells. This is an important question. You should be knowing the answer for this. I think it's there in your MQP. Yeah? Where does the lymphocytes become effector cells, my dear children? Effector cells means the places where they actually go and do the activity. So there are two terms that you need to remember. One is being antigen sensitive. The other one that you have over here is basically antigen sensitive is one thing, and one is being effector cells. Suppose in the case of thymus as well as bone marrow, in the case of thymus as well as bone marrow, this is where the lymphocytes are going to be formed, they mature, and basically we say they become antigen sensitive. That means they know their enemies. But do they do dushum over here? No. They do not do dushum over here. They go to the secondary lymphoid organs such as spleen, tonsils, Peyer's patch, malt, all of these places where they actually show the effect. That is why we say that they are effector cells out there. So in that particular case, your answer for this particular question is going to be what? Your answer is going to be effector cells, only secondary lymphoid organs. This is primary lymphoid organs, cut. This is primary, cut. Peyer's patch and tonsils, none of them are primary, they are secondary lymphoid organs. Bone marrow and spleen, this is also primary, cut. So your only answer for this question is going to be C. So those of you who gave me the answer as C, you got it correct. For those of you who didn't get the answer correct, it's okay, it happens. Okay? Next, antivenom against snake poison contains antigen antibody complex, preformed antibodies, enzymes. What do you think is the answer for this particular question? Antivenom against snake poison contains what? So when you go to the doctor gives you antivenom. Does the doctor say that, hey, you wait, your body will produce antibodies by itself? No, Baba. The doctor will be giving you ready-made antibodies, right? What do you call these ready-made antibodies as? Preformed antibodies. This type of immunization is known as passive immunization. This is known as passive immunization. Okay, Macha, clear? Okay, yes, C, perfect. My dear children are doing super awesome. I'm super proud of you, okay? Now, next question that we have is, by the way, everyone tap on your back because we have come half through of this particular chapter. Now, we just have another half. Let's go. Vaccines work by what? Stimulating the production of antibodies, blocking the entry of pathogens into the body, reducing the inflammation, killing the pathogen directly. So when coronavirus was there, my dear children, everyone was given vaccine. Why? Some people had coronavirus, some people didn't have coronavirus yet. But still they were given what? Vaccine. Why? You wanted to make sure that your body knows that something like this can be coming anytime and you have to be prepared. It's like signaling your body to be ready with all the antibodies against it. Okay, so we wanted the stimulation of production of antibodies. In this particular case, your answer for this question is going to be A, stimulating the production of antibodies. Okay, next thing, 22nd question. Which form of pathogen is used in vaccination? So when you do vaccination, Baba, what are you using? Where are the questions? Where is the question? Yeah. Activated and strong pathogenic antigens, preformed antigens, inactivated and weakened pathogen, hyperactive and strong pathogen. Arey Baba, during corona vaccine, if at all we were given the virus, actually, we would not be standing like this, right? Obviously, you're not going to give strong pathogen out there. You are going to give inactivated and weakened pathogen. This is the answer, right? Perfect. 23rd question. Injection of antitoxin in tetanus confers which type of immunization? Again, during tetanus also you get, my dear children, injection of antitoxin. What does it contain? It's similar to antivenom only. Similar to antivenom. What do you think is the answer for this question? By the way, Rohini, Finaz, Kitson, Sanjana, Kirtana, Sujay, everyone, Mansi, everyone doing super great, okay? And if at all I'm missing out any names, I am super sorry, please forgive me, okay? The names are going like this. And also it will take too much time. So, okay. Rohini, everyone is saying cold. Play says B. B is what? Passive immunization. Exactly. This will be passive immunization. Why, Baba? Because over here also I am giving preformed antibodies. So antitoxin, antivenom, all of them are example of passive immunization. Perfect. Now we go ahead with the next set of questions. Baba, my back's paining. Okay, 24th. Hepatitis B vaccine is produced from, only who have read NCERT knows this answer. Hepatitis B vaccine is produced from, this is from Human Health and Diseases, by the way. Okay, inactivated viruses, yeast, Haemophilus influenzae, Salmonella typhimurium. Which is that particular thing that you will take for basically production of hepatitis B vaccine? Any idea? Ma'am, your explanation is so good. Thank you, love. Thank you so much. Okay, Kirtana says 24B. Okay, 24B, 24B, 24B. Ma'am, you're super duper. Okay, so 24B is what? 24B and D. 24B is yeast. Was it yeast? Yes. Hepatitis B vaccine, Baba. It was produced using recombinant DNA technology. And over here, you had taken the help of yeast. Yeast, yeast, yeast. East or west, you are the best. Okay. Remember? Done? Yes, perfect. Next thing, the most abundant antibody produced against allergens is, so whenever you have allergies, you have some antibodies produced against it as well. What do you call that? What is that antibody? IgE, IgA, IgG, IgM. What do you think is the answer for that? When you have allergy, allergy is for IgE. Allergy is for IgE. IgE. So A is your answer for this particular question. Okay. A is your answer for this particular question. I hope that is clear. 25th, A is the answer. Okay. Okay. Next question. Which of the following cells actively participate during allergy? Now you tell me that. B lymphocytes, liver cells, mast cells and red blood cells. You know about the antibody, but if at all you do not know about the cell which is producing the antibody, not producing the antibody, basically it helps in the working of the antibody. Prajwal, I am not ignoring you, da. Macha, what are you saying, da? Yes, 26C. 26C is what? 26C. Okay, it's mast cells. Exactly, it is going to be mast cells. Listen to me. So I remember it as mast cells. They are very must. Okay, what do have? They have IgE above them. They have IgE on the top of them, and then these are the ones. Basically, it signals. Okay, it signals to the mast cells that hey, mast cell, there is an allergen over here. You have to release two things. One is histamine, and the other one is serotonin. What do? They basically do vasodilation. The blood vessels which are like this, they do like this. Why? Because you want all the other cells to come over here to act on the pathogen, but it is over. Okay, this is dramatic. It should not be needed, but it is there. Okay, so it is mast cells all there. Please remember that. Next question that we have is 27th question. A chemical carcinogen present in tobacco smoke is responsible for. So we say it's a chemical carcinogen. So for those of you who probably thinks that smoking is cool, please need to understand that smoking is not cool. Why? Because it causes cancer. What cancer, Baba? What cancer? 27D. 27D, exactly, lung cancer, and my all dear children know that. It is what? Lung cancer. I hope everyone is against smoking. Yes, do not think it's cool and try it off. Next thing, which of the following approaches is not used for treatment of cancer? So if at all someone has cancer, they will definitely not use this particular treatment for that. Prajwal, I took your name several times, da. Yeah. Ma'am, come to Karnataka, dear. I am in Karnataka. I am in Bangalore. Everyone who is from Vedanta College probably are giving examination in Deeksha campuses. If at all you are, meet me tomorrow here. Okay. 28C. 28C and 28D. Sanjeevani says D. Okay. 28D. Okay, 28D is what? Antiretroviral therapy. Yes, this is not something that you use for cancer. Listen, antiretroviral therapy, that is for viruses. That is being done in the case of AIDS, and over here, what does it happen? Basically, it's not a permanent cure. It is basically a temporary sort of relief that is being given, obviously. Yes. So immunotherapy, surgery and radiotherapy is something that is there for treatment of cancer. Everything else is not. Now, SMAC is a drug obtained from 29th question. Latex of Papaver somniferum. B. Leaves of cannabis sativa. Flowers of datura. Fruits of Erythroxylum coca. SMAC is drug obtained from SMAC, Baba. SMAC. SMAC is what? Heroin. SMAC is heroin. And where do you get heroin from? Heroin, morphine, both of them you get it from one thing only, and that is exactly A. Latex of Papaver somniferum. So Papa has two sons, okay? One daughter, that is heroin, and the second one is morphine. Okay, second one is basically, first one is morphine and the second one is heroin. Okay, we'll talk about it. Anyways, so your answer for this question is latex of Papaver somniferum. Now, the next question that we have over here is tobacco consumption is known to stimulate secretion of adrenaline and noradrenaline. The components causing this could be, so when someone has tobacco, basically smokes up, basically they will have an adrenaline rush and things like that. Okay, what is that something that causes this adrenaline rush? Always remember, when the blood pressure is increasing, it's only increasing because of one guy. And what do you think is the one guy? Someone gave me two answers. 30A, 30C, 30. Why there is a confusion between 30? 30A and C. What do you think is the answer for this particular question? Baba, it is nicotine. Nicotine is the one that causes increase in blood pressure. Okay? Okay? Guys, do not spam. Please do not spam. Okay, just a second. Okay, next question. Heroin formed by acetylation of morphine is commonly called as. I told you morphine comes first, then come heroin, right? All the younger daughters are heroin. So morphine was there and basically it underwent acetylation, and that's how acetylation, and that's how you get heroin or diacetyl morphine. You have the other name for heroin. What do you call this other name for heroin? 31C. C is what? Smack. C is known as smack. So heroin is known as smack. Marijuana is marijuana is extracted from what, my dear children? Ma'am, come to Gurbalga, I will do the arrangement. Oh my god, that's so sweet of you, dear. Okay, 31C. Okay, 32. Shivakumar shower. Okay, C. Kirtana also says C. C is what, my dear children? C is 32A, no. 32A, raisin leaves and flowers of cannabis plant. Listen, cannabis sativa which is there, it is going to give rise to marijuana, hashish, charas, ganja, everything. And each part of that particular plant is going to do that. What do? They are going to affect your cardiovascular system. They are going to affect your cardiovascular system. Always remember that. Okay. Now, charas and ganja are the drugs which affects. You tell me the answer for this. Respiratory, cardiovascular, digestive, the nervous system. What do you think is the answer for this? Ma'am, can you please say there will be question paper will be easy. Okay. I always tell this. Listen. Give your best. Forget about making it easy or difficult. What if I say it's easy and tomorrow it comes out to be hard? You will be disappointed, right? What if I say it's hard? You will still work for it. Work for it. Go. So that is important. Okay, 33B is the answer for this particular question. You are correct. It is cardiovascular system. I told you charas, ganja, marijuana, hashish, everything, they affect the cardiovascular system. Now we go ahead with cocaine. Ayyo. Just a secend. Yeah. So cocaine is obtained from Erythroxylum coca, Papaver somniferum, Atropa belladonna, and Datura stramonium. What do you think is the answer for this particular question, guys? I am doing one questions, all MCQs, dear. I will be talking about other questions also, chapters also. 34A. Okay, Rohini says 34A, Mansi says A, 34A. A is what? Erythroxylum coca. A is Erythroxylum coca. Yes, that is how you get cocaine from. Perfect. Now, cocaine is commonly known as, what do you commonly call cocaine as? Smack, coke, heroin, or cola? Coca-Cola. Cocaine is also known as, Baba, what? 34B, guys, I hope it is clear. A. 33, 35, guys, 35. 35B. 35B is what? 35B is going to be coke. Cocaine is also known as coke, or it is also known as crack. Coke or crack. Always remember that. Next thing, cocaine interferes with the transport of neurotransmitter what? When you say coke is basically, or cocaine is basically a what do you call a drug? What does it usually affect? It affects your central nervous system, CNS. In CNS, it majorly affects what? The secretion or the transmission of what? Acetylcholine, glutamate, dopamine, or glycine. What do you get when you swipe through the mobile phones, my dear children? You only have one thing. What do you think is the answer for this particular question? 36C. Rohini says 36C. Okay. Katijama says it is D. Okay. Asa A. Different answers, my dear children. Rizwana, different answer. Okay, guys. The answer for this question is going to be dopamine. It's dopamine transmission that is being affected. Why? Because of cocaine. Listen, cocaine you get from what? Cocaine you get from Erythroxylum coca. Is it from South America or North America? A question for you guys. Is it from South or North America? Important thing, you should know about it. Okay, and over here, yes, it affects your dopamine transmission. Going ahead with the next question out there, we are going to soon end it. Okay, it's probably the last few set of questions. 37. Which of the following plants does not possess hallucinogenic properties? What do you mean by hallucination? Probably today I am seeing Yasin flying. Tomorrow I will be seeing that Sanjana is also flying. This is known as hallucination, something that is not real. Okay, so you are being asked which one does not possess hallucinogenic properties. The most common plant that causes hallucinogen is basically Erythrochalum coca, that means cocaine and stuff like that. But apart from that, there are two more plants that causes hallucination. What are those? Those are going to be Atropa belladonna and Datura. They are all hallucinogens, my dear children. But our Papa guy, that is Papa where, my dear children, it's not a hallucinogen. So basically this one is the answer. It does not possess hallucinogenic properties. Remember it. Super, super, super important. Ma'am, 100 reached. Yes, dear. Perfect. Okay. I hope it's clear. Yeah. Now, next thing that we have over here is which drug is used? No. Sorry. 38th question. The drug that is being abused by sports person is opioids, barbiturates, cannabinoids, or LSD. Which one is abused? Abused as in people use it too much, such that basically it is going to. Ma'am, I'll come to meet you tomorrow. Are you coming to DVLC? If yes, we'll meet. Guys, just give me a quick second. Otherwise, I think the live might also stop. Just give me a quick second, okay? Yes, perfect. Where did I keep that? Oh my God, just a second. Okay. 39A, 39A is what? 39A is what, Baba? Sports person, it is the drug that is there, it is cannabinoids. For those of you who gave me the wrong answer, bacha, listen to me over here. It is cannabinoid, okay? Cannabinoid as in charas, ganja, marijuana, everything. They are the ones basically which are going to be abused by sports people. Fine? Okay? Jasmine ma'am on fire. Ankit sir, thank you so much. Lots of love. Okay, which drug is used as medicine to help patients cope with depression and insomnia? Insomnia means you're not able to sleep, and basically you're being given drug for that. Okay, such that you have enough amount of sleep out there. So what is that particular drug? Any idea? The one that is there for treating depression as well as insomnia. The answer for that particular question is 39C, Sanjana, Sanju, Kirtana, 39A. Okay, what about others? Yes, I saw Ankit sir. Okay, 39A, 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 39A. Yes, 39A is 39A. Guys, no, 39A is not the correct answer. A is not the correct answer. Listen, look at the question carefully. The drug which is there for treating depression and insomnia. You will be not asked about sedative and a painkiller. If it was sedative and a painkiller, that would have been morphine. Doctors give morphine to people who have underwent surgery. You're talking about depression and insomnia, for treating depression and insomnia. It is BAB. Always remember, BAB stands for what? Barbiturates, amphetamines, benzodiazepines. Okay, it is going to be amphetamines over here because you do not have any other options. Please understand this. Anyone who got it wrong, I hope now it is sorted. Okay, it should be sorted. Perfect. Hassan is also here. Perfect. Now, the last question, the last MCQ question of this particular chapter. A chemical structure is given below. Identify its plant source. What is this particular structure? Where do you get it from? Atropa belladonna, cannabis sativa, Papaver somniferum, Erythrochalum coca. What do you think is the answer for this particular question? JJ ma'am. Yes, dear. Tell her. Tell Krishna Gaming. 40C. 40C is the answer. C is what? Papaver somniferum. Exactly. C is the answer. By the way, do you know what is the structure of? This is the structure of morphine. How do I remember it? I'll tell you. Very easy thing. Morphine comes under opioids, right? Opium poppy. Papaver somniferum is also known as opium poppy. So whatever you get from this, you call them as opioids. So this guy looks like a O. This guy looks like a O. So this is morphine. Okay, perfect. Is this clear? Everyone out there, we have completed human health and diseases, all 40 MCQs within, I think, 40 minutes. 1 minute per question. Perfect. Okay, 1 minute per question. Now we are going to go ahead with the next chapter, which is what? Let me see. So over here, I told you, human health and blood diseases gives you 3 questions. It can be MCQ, it can be fill in the blanks. Anyway, we have covered MCQ, fill in the blanks comes to the easier side. Now we have these chapters that gives 2 marks each. So I think I have tagged already. We will go ahead with. So I've arranged them already. So this has been done. I'll just cross it up and... Ooh, okay, just a second. Okay, let it be there. Human reproduction gives you what? 2 questions. Gives you 2 questions. Okay? Let's go, everyone. Let's go for next chapter out there. That is human reproduction. It will not have that many number of questions as we just solved as of now. And I hope, guys, you are having water. My water bottle is almost empty. Perfect. Ma'am, passed in bio and other subjects, backlog in chemistry, and so when will the result be, ma'am? I'm not sure about the results, dear. I have to check. Okay, 1B. Okay. Scrotum helps in maintaining temperature of testis, which is A, lower than the normal body temperature. It is going to be what? 2 to 2.5 degrees Celsius lower than the normal body temperature. So over here, if at all you see, Baba, just a second. Yeah. Overall, if at all you see, you just have 2 options like that. Let's go ahead. Each of the testes has about B compartments. How many compartments are there? As in how many testicular lobules are there? 250 of them. Here also it's the same thing. Okay. It measures C centimeter in length. What is the length of the testis? Always remember this particular logic. If this is the testis, my dear children, 2 to 3 centimeter is the width and 4 to 5 centimeter is going to be the length. 2, 3, 4, 5. So 4 to 5 is the answer. So which is why your answer for this question is going to be B. It is going to be B. So those of you who gave me different answers, Macha, I hope it's clear now. Lloyd, hello dear. Okay. Okay, Krishna is asking tomorrow 5:30 video. So tomorrow morning I have a live at 5:30. Just for revising the MQPs, nothing else, okay, because yeah, we have been away from it for some time now. So we'll be just revising MQBs as fast as possible. As much as I can cover, I will be covering it, okay? Yes, perfect. Next question that we have right over here is what? Let's go. Okay, spot the odd one out from the following structure with reference to the male reproductive system. Okay, rated testis, epididymis, vasa efferentia, isthmus. Which one is not the part of the male reproductive system? That is the question. What do you think is the answer? 2D, Sanjeevani, Sanjana, Kids, okay, Kids Zone, Sunita, K, Durga Devi. Perfect, perfect. Rohini, Vittal, okay. So your answer for this question is going to be isthmus. Why isthmus, dear? Because isthmus is a part of female reproductive system. You know, fallopian tube. Fallopian tube has what? Infundibulum, ampulla and isthmus. Right? So yes. Which of the following is not a male accessory gland? You just have 3 glands out there. You have been asked which one is not. Seminal vesicle, ampulla, prostrate, bulbourethral gland. Which one is not a male reproductive gland? 2D. I'm talking about the 3rd question. Okay, the live is coming a bit late is what I believe. Yes, 3 answer is B. Vittal says and Kid Zone says. Okay. Kirtana says, Chinnappa is there. Hello, dear. Everyone says B. B is what? Ampulla. Yes. Ampulla is what? The part of female reproductive system. Very easy question. The vas deferens receive duct from the seminal vesicle and opens into urethra as. So, vas deferens is coming from the testis and basically there is a duct over here that is coming and that is known as seminal vesicle. Both of them are going to make a new duct. This new duct which is there, which opens into the urethra is known as what? Epididymis, ejaculatory duct, efferent ductule or ureter. What do you think is the answer for this particular question? 4th question. 4th question. Time duration. This class is going to end by 9:30. Fourth B is the answer. Fourth B is what? Ejaculatory duct. It is going to be ejaculatory duct. Exactly correct. Perfect. Okay, perfect. They are known as ejaculatory duct. Now, the function of myometrium layer present in the uterine wall is two. You have multiple layers: perimetrium, endometrium, myometrium. Perimetrium is the outermost layer, middle layer is myometrium, and the last one, the glandular layer, is the endometrium. So you are being asked, what is the function of myometrium? Read the question carefully. Undergo cyclic changes during menstrual cycle. That is the role of endometrium. Exhibit strong uterine contraction during parturition. Exactly. Fifth answer would be what? Fifth question answer is going to be what? Fifth question answer is going to be B. It exhibits strong uterine contraction during parturition. It is made up of smooth muscles, my dear children, and they do not listen to anyone, just like us. Okay. Now give protection to the uterus, that is perimetrium. Help in implantation process, that is again endometrium. Okay, done. Next question. C. Sixth question. Sertoli cells are found in ovaries and secrete progesterone, adrenal cortex and secrete adrenaline, seminiferous tubules and provide nutrition to the germ cells, pancreas and secrete cholecystokinin. What do you think is the answer for this particular question? What do you think is the answer for this particular question? And everyone, again, I'm reminding you, those of you who have not clicked on the like button, click on it. It reaches, make sure that the video reaches to all the other children out there. Okay, 6C. Okay, sixth C is what? Seminiferous tubules and provides nutrition to the germ cells. Yes, it is found inside the seminiferous tubules, and they are also known as nurse cells. Why? Because they are nursing the germ cell, that is spermatogonia and things like that. Perfect, my dear children. All of you doing super great. Okay, next question that we have over here is basically the cells that secrete testicular hormones, that is androgens, are known as. The male sex hormones are being secreted by what? The male sex hormones which are there, they are being secreted by what? Sertoli cells, Leydig cells, germ cells, or spermatogonia. Seventh B, seventh C. Okay. What is bad, dear? Seventh B. B is what? Leydig cells. Exactly. Luteinizing hormone acts on Leydig cells. Leydig cells produces what? Androgens, that is male sex hormones. LL. Always remember. Luteinizing hormone, Leydig cells. Perfect. Now, eighth question. Seminal plasma in human males is rich in. Seminal plasma basically means all the secretion of your accessory glands, male accessory glands. Okay. So, what is it rich in? Fructose and calcium, glucose and calcium, or basically DNA and testosterone, or ribose and potassium. These are the options that you are having. You have to choose from them. You have to choose from them. Eighth question answer. Farhana says fructose and calcium. What about others? 8A, Kidzone, Rohini, Sanjana, Rupa, HR. Okay, 8A is what? Fructose and calcium. And actually, this is the answer. Fructose and calcium. Who releases fructose? Seminal vesicle. Always remember, three glands are there. Seminal vesicle releases fructose to provide energy to the sperm. Okay, what about prostate gland? Prostate gland is going to release calcium as well as certain enzymes. What about bulbourethral gland? Bulbourethral gland. It is also known as Cowper's gland. It is the one that will be helping in lubrication of the penis. So these are the few things that you need to remember always. Okay, perfect. Now let's go ahead with the ninth question out there. Statement number 1. The process of release of sperms from the seminiferous tubules is known as spermatogenesis. Now sperms will be like, Macha, what are you saying? Okay, the process of release of sperms from the seminiferous tubules is called spermiation, aviation. Remember like that always. Okay, next thing. Statement number 2. Spermatids are transformed into spermatozoa by the process called spermiogenesis. There's a way to remember it. Always remember. Some people forget whether it's spermatozoa to spermatids. Spermatids to spermatozoa. Very easy. Spermatids are like kids. They are immature. They are round globular structure. You want to transform into mature thing, that is spermatozoa, and that is known as spermiogenesis. So I'll say statement 1 is false. Statement 2 is correct. Statement 1 is false. Statement 2 is correct. Your answer is going to be D. Ninth answer is going to be D. Perfect. Awesome. Okay. Next question that we have over here. Dear, I cannot go faster than this. I'll literally die. Literally. Okay. Next thing. In spermatogenesis, if FSH hormone is not secreted from the anterior pituitary gland, which stage is affected? So FSH, my dear children, goes and tells the Sertoli cells to produce what? Factors. To produce factors. Why do you need these factors? We want that the testosterone level is being maintained too much inside the seminiferous tubules, and there should be someone to hold this testosterone. These are the factors, and basically you need it for the whole entire process, but majorly during spermiogenesis, because it is the one where spermatozoa is being formed. So if at all FSH is not there, actually the formation of spermatozoa is being affected. Spermatogenesis process is being affected. Always remember that. So your answer for this question is going to be 10B. Exactly, Jia and Rohini, Mansi, Sanju and everyone. Okay, shots. Perfect. Now, to produce 2000 sperm and 400 ova, how many spermatogonia and oogonia are required? One spermatogonia gives rise to 4 sperms. One oogonia will give rise to only 1 ovum. Always remember that. So, by that logic, if I am saying 2000 sperms are there, so 2000 divided by 4 is equals to what? It gives you 500. It gives you what? 500 what? Spermatogonia. Each of them will give rise to 4 sperms, and that is how you get 2000. Okay, so 500 spermatogonia, you have this option and you have this option. Now what among them? 400 ova have to be produced. Listen, one oogonia releases 1 ova. One oogonia releases 1 ova. So in that logic, if I say 400 ova is there, that means 400 oogonia will also be there. Your answer for that particular question is going to be B. Exactly, Kidzone, Sujay, Rizwana, and people, entertainer, everyone. Perfect. Prithvi, Sanjeevani, perfect. Okay, let's go ahead with the next question then. Okay, diploid number of chromosomes in a primary spermatocyte is 46. What is the total number of chromatids in each of its secondary spermatocyte? For this, you might require time. Think about it and then only answer. Diploid number of chromosomes in a primary spermatocyte is 46. What is the total number of chromatids in each of its secondary spermatocyte? Difficult question. Think about it and then only go ahead with answering it. 12th A, 12th B, okay, Rizwana Farhana says 12th B, okay. Listen to me very carefully. Over here, one cell which is there, okay, I say diploid number, that means 46 number of chromosomes are there. When I say 46 number of chromosomes are there, you need to understand that one chromosome looks like this. And how many of them are there? 46 of them are there. In this particular chromosomes, my dear children, you have this as one chromatid and this is the second chromatid out there. Now when cell division takes place, okay, what is going to happen? The 46 is going to reduce to what? Half of it. During the process of spermatogenesis, primary spermatocyte undergoes reductional division and now it becomes what? How many chromosomes are going to be there? 46 turns out to be 23 chromosomes, half of it. 46 will be turning out to be 23 chromosomes. Listen to me carefully. First, go by this logic. You have 23 chromosomes now, and when we talk about 23 chromosomes, each chromosome looks like this, right? Yes, 23 chromosomes each. Now you have been not asked about chromosomes, you have been asked about chromatids. How many chromatids are there? So if at all you see over here, you look over here, there are 2 chromatids. There are 2 chromatids each. Total number of chromatids in each of its secondary spermatocyte is going to be what? You tell me. You tell me still, if at all you're thinking, initially we had 46, they become half to 23. Now, when I say 23 chromosomes, each of them are having 2 chromatids. 23 into 2 is equals to what? It is going to be a 46 number of chromosomes. Got it? The answer for this question is going to be A. Ma'am, MCQs are from question bank? Yes, these are taken from the question bank. Your answer for this question is A. Is it clear to everyone out there? Okay, perfect. I hope this question is clear, which is why I said it is difficult. Now, the secondary spermatocytes undergo second meiotic division to produce 4 equal haploid what? Secondary spermatocyte is there. It is going to divide further to produce what? First division done. Second division, what do you get? Spermatogonium, spermatids, primary spermatocyte, secondary spermatocyte. What do you think is the answer for this particular question? 13D is what? Secondary spermatocyte. What? Secondary spermatocyte is the one that is undergoing division. What does it form, Baba? Immature ones. Remember kids? Spermatid, small kids, they're immature, they're globular in shape. Always remember that. Okay, B is the answer. 14th question, the process of transformation of spermatids into sperm. Basically, you're asking about spermatids to spermatozoa. What do you call this process? Spermiation, spermatogenesis, spermiogenesis, or basically insemination. What do you call this process as? Guys, please give me the answer as 14A, 14B, things like that. It will be easy for me to understand which question you're solving, right? Okay, 14C. Rizwana says 14C. Okay. What about others? Sanjeevani says 14C and also Rohini. 14C is what, Baba? 14C is going to be spermiogenesis. Yes, this process is known as spermiogenesis. I told you FSH plays a major role over here. FSH plays a major role over here, or basically Sertoli cells plays a major role over here. Now, the next question that we have is release of sperms from the seminiferous tubules is called. What do you call the process of release of sperms? Spermiation, spermatogenesis, spermiogenesis, or spermioteliosis. Okay, what do you call the release of sperm as? Any idea? Sanjana, Shavar, Rohini, and Prithvi, Spurti, Rupa, Jia, everyone doing super great. Okay, JB, Bhagaraj, everyone. Hey, Karnataka seal, hello. So 15th, A is the answer, spermiation. I just told you, aviation. So release of sperm from the seminiferous tubule is known as spermiation. Perfect. Now 16th question, what is the correct sequence of sperm formation? How is the correct sequence of sperm formation happening up over here? Spermatogonia, then spermatozoa, then spermatocytes, then spermatids. Always think the first one is going to be spermatogonia only. Yes. So we have 3 options. We cut off this option. Go ahead, elimination method. The next thing that we have is, always remember, spermatozoa comes at the end. So this particular option is going to be wrong. Over here, look, where does spermatozoa come at the end? Only here. So we have to look carefully in this option now. Spermatogonia forms spermatocytes, primary spermatocytes, right? Secondary spermatocytes, then form spermatids, and then finally you get spermatozoa. The answer for this question is going to be what, Baba? B. It is question B, right? Perfect, Sunita, everyone out there. You guys are super awesome. Next question. In female, oogenesis is initiated during, in female, Baba, oogenesis is initiated during what? Ovulation, puberty, childhood, embryonic developmental stage. When do you think oogenesis is initiated? When does the, what do you call, the process, whole process start to begin? In the case of men, it is in puberty. Okay, what about the females out there? 17th B, who said 17th B? 17D, yeah. 17D is what? Embryonic. Yes, exactly, Rakesh, Kirtana and all. It is embryonic stage. That's when it is going to begin in the case of females. Perfect. Let's go ahead with the next set of questions out there. I think soon it is going to end. Yes. 18th question. Which among the following has 23 chromosomes? 23 chromosomes means half the number of chromosomes. Usually we have 46 number of chromosomes. And if at all we are saying 23 number, then it is half of it. Spermatogonia, zygote, secondary oocyte, oogonia. What do you think is the answer for this particular question? 17th Dia. Okay, perfect children. Sanjana, Vinayak, hello. And Prithvi, Kids Zone, JB Bhagranji, everyone. Babu Rao. By the way, everyone is trying, right? JK News. Okay. Mansi says 18C is the answer. 18C is about Baba. 18C is secondary oocyte. Secondary oocyte, will it have it? Yes. Always remember, from the secondary stages only, you will have half the number of chromosomes. Until then, everything, primary oocyte, primary, whatever is there, oogonia, spermatogonia, everyone will be diploid in nature. Here, this guy will be haploid in nature. Perfect. Whether it's secondary oocyte, whether it's secondary spermatocyte, it is going to have half the number of chromosome. 19th question. Gonadotropin releasing hormone is a hypothalamic hormone which stimulates the pituitary gland to release what? Pituitary gland releases what? When GnRH comes and lands on the pituitary, what is going to be released? Listen, the name is written, gonadotropin releasing hormones. It asks the pituitary to release gonadotropins. What are the gonadotropins, my dear children? Only 2 gonadotropins are there. Right? Only 2 gonadotropins are there. 19th A is the answer. Yes, it is LH and FSH, luteinizing hormone and follicle stimulating hormone. Perfect. Let's go with the next questions that is out there. 28. Identify the wrong pair with respect to menstrual cycle. Come on, we have done it so many times. Ma'am, humans are health and disease industry. Yes, you need to write. Okay. Formation of corpus luteum 15th to 28th day. Corpus luteum, my dear children, happens after ovulation. Okay, so when the graphene follicle ruptures to form the secondary oocyte, whatever is left behind is known as corpus luteum. And yes, it is going to be there from the 15th to 28th day. And this is the guy that releases the progesterone. So when this guy is there, obviously progesterone will be increased. So secretion of progesterone 15th to 28th day, exactly. Regeneration of the endometrium. Listen to me. Endometrium falls down when on the 1st to 5th day when you have menstrual cycle, right? 1st to 5th day you have menstrual cycle, that's when it falls down. From 5th to 13th day, because on the 14th day you have ovulation, 20-day cycle is there, half of the cycle, 14th day you will be having ovulation. Before that you need to regenerate the endometrium a bit. So from 5th to 13th day regeneration happens. This is correct? Yes, correct. Disintegration of endometrium from 5th to 13th day, no baba, it happens on 1st to 5th day. 1st to 5th day it is going to disintegrate. Okay, so your answer for this question is going to be what? 20D. Okay, 20D is the answer. Disintegration of endometrium is not this. Perfect. Now the corpus luteum secretes large amount of progesterone, which is essential for the maintenance of. What is corpus luteum? Why is it releasing lots of progesterone? For what? For maintenance of what? Perimetrium, myometrium, endometrium, cervical canal. What does the baby need? So when the embryo is formed, baba, there should be someone to nourish it, right? So it should be the endometrium. It should be someone. Endometrium, right? So 21C is the answer. 21C is the answer, and that is endometrium. Perfect. Next question. Progesterone hormone is secreted by. Progesterone, which is the hormone, it is secreted by, baba. I told you right now. Testis, pituitary gland, corpus luteum, graphene follicle. Who releases progesterone? The one and only. 22nd question. The one and only. Ayyo. I feel so happy that you guys are attempting. Super proud of you, okay? Okay, so 22C. 22C is what? Corpus luteum. Yes, corpus luteum is the one that releases progesterone. That is absolutely correct. Now, which of the following statement is true? You have to find it out, okay? LH surge induces menstruation. Baba, LH when it surges, does it induces menstruation? No, it induces ovulation. This is wrong. You have to find what is true. Now, when true and false question come, first of all, find what is written over here. Sometimes we try to find what is false. Okay, so everything will be false. We will like, amma, what is happening over here? Okay, regression of corpus luteum increases progesterone. Baba, corpus luteum, if it is going down, how can progesterone go up? Both of them are simulated. Corpus luteum is the one that maintains progesterone. So this is wrong. No. If this guy is going less, how can it increase? This is a wrong statement. Diploid egg is formed after second meiotic division. The egg is crying at a corner now. How can the egg be diploid? It is always haploid in nature. So diploid egg is formed after second meiotic division. Wrong, baba. It is going to be wrong. Now, last one. Embryo differentiates from the inner cell mass of the blastocyst. So blastocyst is the embryonic stage when the embryo finally gets into the endometrium, and you have two layers, trophoblast and you have the inner cell mass. Trophoblast is going to help in the formation of chorionic villi, later placenta and things like that. The inner cell mass is the actual one which forms the embryo proper. So we say embryo differentiates from the inner cell mass of the blastocyst. This is actually the correct one. This is the only correct statement. Done? Done, ma'am. Okay, now 24th question. Okay, 24th question. Match the items given in column 1 with those in column 2. Okay, proliferative phase, secretory phase, menstruation, and then you go like this. First of all, easy way to attend to your answers. A and C, sort it out first. Proliferative phase means what is proliferating over here? Endometrium is slowly beginning up. It is proliferating, and by this particular time at the top, follicles are also regenerating. The follicles will be producing estrogen. That is why the endometrium proliferates. So I'll say follicular phase. A goes with 2. A goes with 2. I just have one option, and basically with that I got my whole answer. Right. 24th answer is what, macha? C. Okay, secretory phase is basically luteal phase also. Why? In luteal phase, corpus luteum is there. Progesterone is being released. Progesterone maintains the endometrium. It makes the endometrium release certain very important things also, and that is known as secretory phase. Menstruation, baba, it is known as breakdown of endometrial lining, and your answer is done. 25th question. When does the second meiotic division occur in the secondary oocyte? When does the second meiotic division occur in the secondary oocyte? At the time of birth, at the time of fertilization, at the time of puberty, at the time of ovulation. Baba, secondary oocyte is there. It has to divide and form what? Ovum and the second polar body. When does it do the division? During ovulation? No. During birth? No. During puberty? No. What are we left with? At the time of fertilization, not after fertilization, okay? For fertilization, you need the ovum. So always remember, before the process happens, okay? Before it's just about to fertilize, you will get it. So answer for this question is going to be B. Clear? Okay, Sanjana, Sanjeevani, Shower, Dilip Kumar, everyone, perfect, good too. Okay, Hassan. So, guys, it's B, okay? I hope you're solving 25th question. Yes. Perfect. Now we go ahead with the next thing that is there. The secondary oocyte after ovulation is covered by non-cellular layer. Secondary oocyte, when it is released, Baba, it is covered by a layer. What do you call this non-cellular layer as? Cumulus ophorus. It feels like I'm speaking Spanish. Corona radiata, zona pellucida, and cortical layer. What do you think is your answer for this particular question? Babu Rao says B. I think for the previous question, yes. 26th question, where are we? Sanjeevani says C is the answer. Okay. Super clear. Thank you so much. Okay. The answer for this question is Baba, zona pellucida. Okay. Zona pellucida is the answer, C. So secondary oocyte makes its own layer, and that is known as zona pellucida. It is not having any cells out there. Very important. Corona radiata is also being found, but not by secondary oocyte. It is being found by the other cells of the granulosa cells. Perfect. Clear? Yes. Now, come on. How many questions are there? Okay. Few more. We can do it. Okay. Choose the correct option from the table given below. Okay. It is formed by, what is this particular layer? First of all, what do you call this particular layer? Zona pellucida. It is formed by what? We just studied about it right now. It is formed by secondary oocyte. So two options are there. Cut off these three options. Okay. Sorry, two options. Now, what are we left with? When does it dissolve? Now, this is a confusion for every one of us. Okay. When does it dissolve, macha? Before fertilization, after fertilization? Before fertilization, after fertilization. 27, the answer is D. Yeah. Is it D? Exactly, yes, it is D. Listen to me very carefully. So when the sperm comes in, it makes a small gap in the zona pellucida. It's not like dissolves all of it. So you should not be thinking that before fertilization it happens. No. After fertilization, basically after the egg and the sperm fuses, slowly the zona pellucida layer will be going on. Until then we need it. Why? We do not want any sperm to enter into it, right? So that is why. So it is after fertilization. Your answer for this question is going to be D. Okay, I hope it makes sense. Which of the following hormones is produced only during pregnancy? Only during pregnancy. HCG, LH, estrogen, FSH. HCG stands for human chorionic gonadotrophin. HCG stands for human chorionic gonadotrophin. What do you think is the answer for this? Produced only during pregnancy. Arey Baba, we have luteinizing hormone, we have estrogen, we have FSH. This is all normal things. These two are pituitary hormones, this is ovarian hormones. Which one is not? This is going to be HCG. Okay, this is something that is released only during pregnancy. Okay, the dash hormone is secreted by the ovary in the later phase of pregnancy. Towards the end of the pregnancy, there is a hormone that is being released by the ovary. What is this particular hormone known as, Baba? 29th question. 29th question. No, don't close. Come on. Yes. Okay. Arey. Just a second, guys. If you do not want your teacher to die because of heat, you would have to wait for a second. Yes. 29C. 29C is what? 29C is relaxin. Yes. The ovary say relax macha, baby has to move out, you give some space and all. No. So relaxin is for relaxing the pelvic ligaments out there. Okay, done. Now which of the following hormones is not secreted by the human placenta? Not secreted by the human placenta. There are 4 hormones secreted by it: HCG, HPL, estrogen and progesterone. Which one is not? It is obviously going to be LH. LH is not something that is going to be secreted by the what do you say, the placenta. Okay, so your answer for this question is going to be D. Perfect. Yes. Someone is saying C. Okay, fine. 30th question. 30th D. Yes. Mansi. Yes. Purvika. Perfect. Now, morula is a developmental stage. Morula is a development stage between what? Between zygote and blastocyst, between blastocyst and gastrula, after implantation, between implantation and parturition. Listen to me carefully. You have the zygote. The zygote undergoes a process that is known as cleavage. Okay, basically it divides. It was a single cell. It divides into two into four, and that's how it goes. So we now call this as embryo. Whenever it is not a single cell anymore, we will now call this an embryo. And in embryo, you have multiple stages. The first stage that you have to remember is morula. Morula is basically cell, basically an embryo up to 8 to 16 cells. 8 to 16 cells. Okay. After morula comes what? Basically so many things, but majorly blastocyst is going to come. And blastocyst is going to undergo implantation. So can I say between zygote and blastocyst? Yes, I can say that. After blastocyst, gastrula and stuff like that is going to be formed. So 31st answer is going to be Baba A. It is going to be A. Perfect, Sanjana, Adar Sanjana, Purvi, Spandana, and Mansi. What about the guys out there? Where are all the guys? Perfect girlies, going on really, really strong. Okay, the next question that we have over here is, yeah, 32. Which one of the following is not the function of placenta? Facilitates removal of carbon dioxide and waste materials from the embryo. Secrete oxytocin during parturition. Facilitates supply of oxygen and nutrients to the embryo. Secretes estrogen. I told you it releases 4 hormones, and under that comes estrogen. But oxytocin basically is not being released over here. So this one would be the thing that is going to be wrong. Agreed? Agreed? Ma'am, I think you're tired. Definitely, I'm super tired. I'll be taking rest. Post this class has been done. Okay. Done. Guys, B is the answer for this particular question. Let's go. Next question. During pregnancy, the fetus develops limbs and digits. During pregnancy, the fetus is going to develop limbs and digits. After how many time? After 3 weeks, after 8 weeks, after 6 weeks, after 9 weeks. What do you think is the answer for this particular question? During pregnancy, the fetus develops limbs and digits after what? Yes, oxytocin is released by mother's pituitary. Exactly, Hibs. Okay. Ma'am, not D. D is what? No, it's this. Okay. Implantation, how can that be happening? Implantation is basically blastocyst, right? Yes. Your answer is going to be C. Listen to me, guys. First month heartbeat. Okay, 1 month has 4 weeks. Now 2 months, the second month you have limbs and you have digits. So basically I'll say 2 into 4 is basically 8 weeks. Your answer for this question is going to be B. It is going to be B. 33 answer is B. 33, why 6 weeks, Baba? 33 answer is B. Okay, always remember. Why C? Why C? I see people saying C after 6 weeks. 6 weeks means what? Basically 1 month is done, and then no. After 8 weeks, okay, 2 months, 2 proper months. 8 weeks. Now, the first movements of the fetus and appearance or hair on its body is usually observed during, this is one of my favorite. The baby says, until now I cannot move. Please let me move. I want to dance, and also I want to show some style. So, first hair and first movement basically is found in the 5th month. In the 5th month, my personal favorite month out there. 5th month out there. Okay. 34th answer for this question is going to be B. Perfect. 33, explain 33 question. Fetus develops limbs and digits, hands and digits, after the end of 2 months. After the end of 2 months. Every month has 4 weeks. 2 into 4 is equals to what? 8 weeks. No. So after 8 weeks it is going to happen. Done. Got it? Perfect. Next thing that we have is last 2 questions, guys. During embryonic development, embryo's heart formed at the end of first trimester, second trimester, first month, third month. Embryonic heart, my dear children, the first thing when you die is being checked is your pulse rate. That means basically your heart is beating or not. Similarly, the first thing when you're born is being checked is your heartbeat, and that has been done after the end of first month. 35th answer is going to be what? Okay. 35th answer is going to be what? Baba? C. 35th is going to be C. First month is the answer. Cool. Dear, I do not have the PDF, Vinayak. Okay, next thing, signals for parturition originate from. Who gives the signals for parturition? That means baby wants to move out, baby wants to move out. Who gives the signals for that? Basically, both placenta as well as the fully developed fetus. Yes, this is known as fetal ejection reflex, and it is being done both by placenta as well as the fully developed fetus. The fetus tries to move out. When it tries to move out, it basically activates the stress receptors out there in the cervix, and that gives a signal. Apart from that, basically the baby which is there, the fully developed fetus, also releases some hormones, and these hormones goes to the placenta, and placenta also basically makes some changes out there leading to the fetal ejection reflex. Okay, so your answer for this question is going to be A, both placenta as well as the fully developed fetus. Yes, Purvi, tell me. Ma'am, for model and how many marks can we get? Literally, I cannot do that. Ma'am, will you do live classes? Definitely will be doing that. Yes? Okay. Going ahead with the next chapter. Guys, first of all, big tap on your back because you are doing super awesome. You are fantastic. You are lovely children. Okay? Perfect. Shall we go ahead with the next question? After completing syllabus, I'm not feeling confident. Do some hot question from the question bank. See if you're able to do it or not. If not, then try it up. And by the way, please make sure you rest. Okay. Perfect. We are going ahead with the next chapter. Microbes and human welfare gives you 2 marks each. Okay. Let's go. So thank you, Mansi. Okay. So the first question that we have over here, before doing that, we need to do some... Okay. Okay, guys. Propionibacterium shermani is used for the production of Roquefort cheese, Swiss cheese, bamboo shoots, and ferment fish. What do you use for? Your motivation. Thank you. Because we had the best teacher. Oh, that's so sweet of you. Okay. Ma'am, can you do MCQs of all the chapter? Dear, sadly. Just a second. Yes, perfect. So sadly, I may not be able to do that because the time is up. Tomorrow morning I have to come by 5:30. Okay, so it's been 2 days since proper sleep is not happening. Okay, and it might take too much time. Okay, B is the answer. Yes, B is the answer for this question. That is Propionibacterium shermani. It is shermau. Why? Because it is releasing so much amount of CO2, which creates big pores. Swiss cheese, the one in Tom and Jerry and all, you might have seen a cheese with big holes, right? So basically, this is Swiss cheese. Okay. Penicillium notatum is what? A bacteria, virus, blue-green algae, or a mold? What do you think is the answer for the second question? Penicillium notatum. Penicillium notatum is basically what? Is basically what? Any idea? A. Sneha, it's there in the WhatsApp channel. And you're most welcome. Thank you so much, Jia. Okay, 2D is what? 2D is mold. Exactly. So mold is basically, what do you mean by mold? It's a fungus. Penicillium notatum is the one that releases penicillin, and penicillin is what? It's an antibiotic. So this mold is killing the bacteria by releasing antibiotic. Okay. Alexander Fleming discovered penicillin while working on what? Staphylococci, Streptococcus, E. coli, and Salmonella. This is where most of us might do mistake. So please do not do this mistake. Staphylococci, Streptococcus, E. coli, Salmonella. Where does, or basically Alexander Fleming used what for the making of penicillin? Any idea? When? How did he discover it? Srinidhi, thank you so much, dear. Okay, 3A is what? 3A is Staphylococci. Perfect. The answer is Staphylococci. He was growing Staphylococci. One day he's like, Macha, I forgot to wash the dish. Tomorrow there will be so many Staphylococci. But day after tomorrow when he came, he saw that something was not allowing Staphylococci to divide, and that was penicillin. Well, always remember, if at all you're a bit confused between this and this, here you have L. Alexander also has an L. Okay. Now, which one of the following alcoholic drinks is produced without distillation? You have been asked without distillation. That means, what is the process of distillation? Distillation is basically when you are letting all the water content go away. That means over here the concentration of alcohol is going to be too much. So whenever you get things from distillation, that means you get concentrated activated alcohol form. Without distillation means water content is still there, it's still fine. Okay, it's still fine out there. So your answer will be, which of the following alcohol drinks is produced without distillation? The ones which will be having less alcohol content. Obviously over here whiskey, rum and brandy is there. The answer would be wine. The answer would be wine. How will my children know? My children do not didn't call these things. But yes, my children know this. Why? Because we have studied it, right? Next question. I hope just because of that. Okay. Now because of the presence of which vitamin curd is considered nutritionally richer than milk? Which vitamin is there that makes the curd nutritionally richer than milk? Any idea? Okay. 5D is what? Vitamin B12. Exactly. In the case of milk to curd formation, you have lactic acid bacteria, and this lactic acid bacteria basically adds some vitamin B12 also. This is why your answer is going to be D. Bioactive molecule cyclosporin A is produced by fungus, bacteria, virus, protozoa. Cyclosporin A, Baba, is produced by Trichoderma polysperm. Now you tell me whether it is fungus, bacteria, virus or protozoa. What do you think is the answer for this particular question? Yes, definitely Vinayak, I'll definitely try so. D. Take our names. Purvi, yes. Okay. So, sixth question you have been saying, right? Sixth question, Karnataka, Asil, as well as Lakshmi, give me the correct answer. Rizwana, give me the correct answer. Spandana, Sudha, Sanjeevni, and okay. Mansi, okay. A is what? Fungus. Always remember, Trichoderma polysperm is the fungus that releases cyclosporin A, which is a immunosuppressive agent. Seventh question, which microorganism is useful in the production of citric acid? Aspergillus niger. Okay, Acetobacter aceti, Penicillium notatum, Aspergillus niger, plus Clostridium botulicum. What do you think? Which one is the one that basically helps in the production of citric acid? Any idea? Ma'am, you're fired. Thank you, Kid Zone. Okay, seventh, C is the answer. C is what? Aspergillus niger. Remember, Aspergillus niger is a fungus. Clostridium botulicum is a bacteria. It creates butyric acid. Penicillium notatum again is a fungus. Acetobacter aceti is a bacteria that forms acetic acid. Okay, activated sludge is a sediment deposited in settling tank, sedimentation tank, aerobic tank, anaerobic sludge digester. What do you think? Where do you get the activated sludge? What do you think is the answer for this question? Sanjana, Mansi, Shower, and Sanjana, Sudha, Spandana, everyone. Sangeeta, perfect. Guys, next question. 8A, Sanjeevani says 8A. 8A is what? Settling tank, exactly. Activated sludge is what? Basically flocs. Okay, that is the aerobic bacteria plus the fungal filaments. They are being formed inside what? This settling tank. So your answer for this question is going to be A. It is going to be A. Perfect. Yes, settling tank, do not forget it. Now, yeast is used in the production of bread and beer, cheese and butter, citric acid and lactic acid, lipases and pectinases. Where do you think cheese is going to be produced? Where do you think cheese is going to be produced? What am I saying? I'm so sorry, children. Brain is not braining. Okay, yeast is produced in the production of what? 8A. Perfect, Lakshmi, Sanjana, Spandana and all. Ma'am, next thing, Sneha, ma'am, your teaching skill is super. Thank you, dear. Okay, 9th A, Purvi says A, Shower says A. A is bread and beer. Exactly. Saccharomyces cerevisiae is known as brewer's yeast as well as baker's yeast for this particular reason. Okay, perfect. Let's go ahead with the next set of questions out there. Biogas is a mixture of. Biogas, Baba, is made up of what? It is not just CH4, that is methane. It is also H2S and also CO2. In that case, what do you think is the answer for this particular question? Here you do not have H2S, right? Here you do not have H2S, so obviously you have to go with H2, hydrogen. Otherwise you would have gone with H2S. 9th A, Indira, Indu, perfect. Srinidhi, ma'am, have a Kit Kat break. Definitely wanted to have, but after this, okay? Dilshad, you're most welcome, champ. Okay, 10th A, Lakshmi, Purvi, okay. 10th A is what? CH4, CO2 and H2, exactly. So CC will be there, okay, and one more guy is there, H2S or H2. So basically this is the answer. Now 11th question, mesh-like structure consisting of bacteria associated with fungal filaments. Bacteria, fungal filaments, all of them are like hand in hand. What do you call this structure? What do you call this as? Primary sludge, activated sludge, floc or anaerobic sludge? What do you call this as? Mesh-like structure which is there. What do you call this as? 11th C, Sunita says 11th C. C is what? Flock. Yes, you call this as Baba flock. Okay, you will call this as flock. Vijay Chandan Kumar, my dear child, I cannot ignore anyone right now. I mean, I'm possibly ignoring everyone right now because the chat is going like that, but I'm literally trying my level best. Okay, Rizwana, Sanjini, Mansi, Purvi, everyone, Travan, Mantya official. Okay, so the answer for this question is going to be C, Macha. Next one, the primary sewage treatment involves anaerobic bacterial activity, sludge digestion, filtration, sedimentation. Aerobic bacterial activity, listen, is the first physical step that is out there for the treatment of sewage. At this particular time, you have simple steps, and that is basically what? Filtration and sedimentation. You just have filtration and sedimentation. Whatever is floating, you remove through filtration. Whatever is sedimenting, you remove through sedimentation. Okay, done. Okay, perfect. Let's go ahead with the next question. 12th C, Sanjeevani, Sanjana, Manjunath, everyone, Rizwana, Spandana, Mansi, Sharan, and Vijay. Everyone, kids zone, perfect. Next thing, 13th question. Match column 1 with column 2 and select the correct option using the codes given below. Citric acid, cyclosporin A, statins, butyric acid is given. Now, citric acid, we just studied right now. Citric acid is produced by Aspergillus niger. Okay, now butyric acid. Do not go ahead one by one, go the next one. Butyric acid is basically found by Clostridium botulicum. So, A goes with 3, I just have 2 options. I will cut off this option, 3 options, sorry. And D goes with 2, with 2, I have basically 2 options. So these are the things that I have to be careful for. Cyclosporin A is made by Trichoderma polysporum. B goes with 1, and basically I've got my answer. The answer to this question is going to be what? It is going to be what? Navneet, dear, if you need any, I mean, I cannot go faster than this, dear. Ma'am, what is your name? My name is Jasmine Joy. Okay, yes, B. Answer is going to be B. Guys, are you doing B only, or there is some different question? And statin is by Monascus purpureus. Okay, 13th B, perfect. Next question that we have over here is what? Which of the following is in sewage treatment removes suspended solids, as in the ones which are like in the solids, and basically we want to remove them. So which of the process is it? Secondary treatment, primary treatment, sludge treatment, or tertiary treatment? What do you think is the answer for this particular question? Ma'am, we will tell the answers. Yes. Please. Go ahead. Microsoft. Ma'am, should I just do model paper and preparatory for 15 theory? If at all you have not done anything now, then probably yes. But if you can, go watch the link that is given in the WhatsApp channel. They have given you something, resources. Okay, you can join the WhatsApp channel for sure. It will help you. Okay, B is what? B is going to be primary treatment. Exactly. Listen, you want to remove the suspended solids, no? So there is one way, sedimentation, where all the solids, rock, soil, everything, grit, everything comes down. So you can remove through that. Okay, so over here B is the answer. Now 15th question, during sewage treatment the gas produced in anaerobic sludge digester include. Anaerobic term is given, that means there is no oxygen over here. In this particular condition, what gas is going to be produced? I always say anaerobic condition, that means one type of gas. And what is it? Tell me. Hydrogen sulfide, nitrogen, methane, oxygen, hydrogen sulfide, methane, hydrogen sulfide, and carbon dioxide. Hydrogen sulfide, methane, and sulfur dioxide. What do you think is the answer? How to join the channel? So listen to me carefully. If this is your video that you're watching, click on the more over here. You get first link out there. Click on that particular link. You will see lots of messages out there. Yeah? Yes. Perfect. So 15th C is the answer. Is it? Sanjeevani says 15C, Shavar says, Spandana says, and Dilip Kumar also says it is C. Exactly. Methane CH4, carbon dioxide CO2, H2S, that is hydrogen sulfide. Got it. Perfect. Mycorrhiza is involved in absorption of dash from the soil and passes it to the plant. Mycorrhiza is involved in absorption of what, Baba? Mycorrhiza is a combination of fungus as well as the plant roots. And what does it absorb? Mansi, everyone, okay? Dilip Kumar, Shavar, Spandana, Mantya, Shiva, okay. 16th B is the answer. 16th B, Rizwan also says 16th B, okay. Mansi, Hassan, everyone says B, that is phosphorus, exactly. It goes deep into soil to trap phosphorus. Mycorrhiza, phosphorus, not potassium, please do not remember like that, okay. It is phosphorus only. Next question, which of the following is not an advantage provided by mycorrhiza to the plants? Basically, fungi helps in giving phosphorus. Apart from that, there are so many other things which it does. But which one is not an advantage is being asked? Resistance to root-borne pathogens, does it provide it? Resistance to root-borne pathogens, my dear children, A. Does it provide it? Sneha, you're most welcome, dear. Hire Gowda, you have to go ahead with some extra questions if at all you can try. Okay, ma'am, why bio is this hard? It's easy. Do not make it hard in your head, okay? Okay, 17B, Sanjeevani, Sanjana, Sunita, Mantya. Okay, B is what? Increase in yield quality. So listen to me. Resistance to root-borne pathogens, yes, is provided by the fungi. Tolerance to salinity and drought, yes, that is also provided. Overall increase in plant growth and development, that is also provided. But yield quality, that is not something that is provided over here by the fungi out there. So your answer for this question is going to be this. Okay, the next question that we have is 18th question. The microbial biocontrol agent for butterfly caterpillar is butterfly, oh my god, butterfly caterpillar is controlled by which agent, guys? Bacillus thuringiensis, Saccharomyces, Cyanobacteria, Staphylococci. What do you think is the answer for this particular question? Okay, Sanjana Hulagappa says 18A. Okay, slowly so it will come. Bio is easy, okay. So, Shweta, Shiva, Siddharaya, okay, Siddharaj, sorry. 19th question, guys, 19th question. Thank you so much, Vinay, that's so sweet of you. 19th question. Are we solving? Sorry, 18th question only we're solving. I'm so sorry. Its answer is what? Bacillus thuringiensis. Your answer is going to be A. Perfect. It is going to be A. Thank you so much, guys. Next question. Which of the following is a bacterial biofertilizer? That means basically this particular organism increases the fertility of the soil, and you do not need any other fertilizer, chemical fertilizer out there. But it should be bacteria. Annabena, Nostoc, Rhizobium, Oscillatoria. What do you think is the answer for this particular question? Please understand it is bacteria that is being asked. Okay, ma'am, it's cute when you started. Oh my god, that's so sweet. That's the first time ma'am was not feeling well today but studied. Now I'll revise, but MQP tomorrow morning. So will you just do MQP morning or include anything else too? Alana, it will be just MQP and I'll try to do it as fast as possible. You know my speed, right? Yes. Sanjeevani says the answer for this is C. Okay, Shiva Shankar also says, Yash also says it is C. Sanju, C is what? Rhizobium. Listen to me very carefully. There is cyanobacteria, there is bacteria. Two different things are there. Okay. Cyanobacteria are the ones that can prepare their own food. For example, Annabena, Nostoc and Oscillatoria. Always remember ano. Okay, ano. Instead of you know, ano. Annabena, Nostoc, Oscillatoria, they are cyanobacteria. Rest everything is basically bacteria. Rhizobium, Acetobacter, Azospirillum, everything. Okay, which of the following is not a biofertilizer? Which one is not a biofertilizer? Agrobacterium, Rhizobium, Nostoc and Mycorrhiza. You know the answer, don't you? Okay, you know the answer, don't you? Okay, Purvi, Mansi, Siddharaj, Yash, Indra, and I might be reading some wrong names as well. Sigma Mama, oh, thank you, dear. Okay, what do you think is the answer for this question? 19th. JJ Speed from India, oh my god. 20th question. 20A, it's Saiba says, hello. So 20A is what? As agrobacterium. Yes, agrobacterium which is there, it is not a biofertilizer, Baba. It is a biocontrol agent, right? Right. Now going ahead with the next question, 21st. Which of the following statement regarding phlox is incorrect? Incorrect is what you have to find. Don't find the correct one. They are the masses of bacteria associated with the fungal filaments. That is correct. Bacteria, fungal filaments, they are close together. Okay. Produced in the aeration tank. That is also correct. When you give air, then only aerobic bacteria can survive. And aerobic bacteria and the fungal filaments are the ones that form phlox. Correct. They consume major part of the organic matter. Yes, they are the ones that is going to consume the organic matter and lessen the pollution. That means one more statement is there and that will be the answer, right? So sorry, I gave the answer for this question, and that is what? Formed by useful anaerobic microbes. My macha, you only think, Baba. Flocks which are there, they're produced by aerobic bacteria, not anaerobic bacteria. Here it is mentioned about anaerobic bacteria. Do you think that is correct? No, right? So 21st answer will be D. Forgive me, I gave the answer already. Okay. Next one that we have is 22nd question. Compare the statement 1 and 2. Okay, let's compare. Statement number 1. Mycorrhiza is a symbiotic association of algae and a fungi. I am not saying anything. Statement number 2. They absorb nitrogen from soil and pass it to the plant. Tom. Tell me the answer for this. Statement 1 is true. What is true? What is not? Okay. Srinidhi, Roll Sun Lobo. Okay. Abhishek, hello. Yash. Hello, lots of children are there. I'm not able to talk to you. I'm literally sorry. If at all, probably tomorrow, if it wasn't your examination, I would have spoken to all of you. Okay. Ma'am, I'm regretting taking bio over CS. Don't say that. Okay. 22B, 22D. Sanjini says 22D, 22A. Mansi says, why there is a difference in opinion? Why there is a difference in opinion? Why? Because you have not read the question carefully. Mycorrhiza is a symbiotic association of algae and a fungi. Who said that? It is a plant root and the fungi. This statement, it's wrong. It's lichen you're talking about. It's not mycorrhiza. Here it's mycorrhiza, right? So it's wrong because this is lichen. Statement number 2. They absorb nitrogen from soil and pass it to the plant. The mycorrhiza right now will be like, because nitrogen is not the one, it is phosphorus, right? So both the statements are wrong, Baba. Do you have any option like that? Yes. D. Both the statements 1 and 2 are false. So who gave me the correct answer? Sanjeevani gave me the correct answer. Purvi gave me the correct answer. And Purvi. That's all. Spandana also gave me the correct answer. Sanjana gave me the correct answer. Okay. Guys, I hope this is clear. Yes? Yes. Perfect. Okay. Now, next thing that we have over here is the 23rd question. What does it say? Select the correct statement from the following. Biogas is produced by activity of aerobic bacteria on animal waste. Okay. B. Methanobacterium is an aerobic bacterium found in the rumen of the cattle. Okay. Biogas, commonly called gobar gas, is pure methane. Activated sludge is settlement tax of STPs, sewage treatment plants. Okay. Is a rich source of aerobic bacteria. What do you think is the answer for this question? You have to find which one is the correct statement from the following. Let me see who can. We can score 15 out of 15 from this. No. Because how can I guarantee that? What if they are giving you questions apart from the question bank? Aksa, so this is honesty and this is what I am telling you. Ma'am, how is your back pain? My back pain is literally bad, but yes. I personally don't like biology, but hats off to your teaching, ma'am. Thank you, Rishi. Thank you so much. Okay. Guys, what do you think is the answer for this question? 23C. Sunita says 23C. Jia has C. Okay, Shweta says D. Okay, multiple answers. What is the answer for this question? There is only D. Why? Biogas is produced by activity of aerobic bacteria. Wrong. It is anaerobic bacteria. Biogas, anaerobic. Wrong statement. Methanobacterium is an aerobic bacterium. Wrong. It is an anaerobic bacterium. That is the one that that makes biogas. Biogas, commonly called gobar gas, is pure methane. Papa, CO2, NH2 is crying in the corner. Okay, it is not made up of just methane, but also because of them. Activated sludge in settlement tanks of STP is a rich source of aerobic bacterium. Correct. Settlement tank is correct. Rich source of aerobic bacteria, that is also correct. So the answer for this question is going to be D. Okay, it is going to be D. Done. Perfect. That chapter is also done. Give a big tap, huge tap on your back. Everyone. Every single chapter when you're done, you have to do that. That's like a motivation. Ma'am, people are increasing, is it? Yes, they have been increasing some time now. By the way, everyone who have not clicked on the subscribe button, please click on the subscribe button, machas, and what? Click on the like button. Yes, and everyone who is watching this video, remember me. Okay, remember me if your examination goes bad, and also if your examination goes good. Okay, so usually it's the other way people say, but I'm telling you in both the ways. Please come and tell how was your examination, post your examination is being done, and I'll be praying for all of you, okay? Yes, work really hard, give your best, and be calm. Ma'am, new subscriber, is it? Thank you so much, Srinidhi. Okay, next thing that we have is biotechnology, principle and processes. Again, going to give you 2 marks. Okay, again going to give you 2 marks. We will zoom it in, and this time we'll change the color. Let's change the color to lavender. Okay. Which of the following is not a feature of plasmid? Extra chromosomal, circular structure, single-stranded, independent replication. Which one is not the, okay, which one is not the feature of plasmid? Thank you so much. Oh, that's so sweet of you, Rupashri. I love you too. I love everyone out there. When you will be live again tomorrow morning at 5:30 a.m. for all the MQPs, I'll do it very fast. Yes. Sanjana, thank you so much. Everyone, Spandana, Yash, Sunita, Rupa, Aksa, okay, Chinmay, Vinayak. Okay. Guys, lots of love. 1C. C is what? C is single-stranded. Listen, your DNA which is there, I'm not liking it. DNA which is there, it is going to be double-stranded, not single-stranded over here in the case of plasmid. So rest everything is correct, but this is wrong. The procedure through which a piece of DNA is introduced into the host bacterium is known as. Remember, do not mess around here. The procedure by which a piece of DNA is introduced into the host bacterium. We are putting it inside the host bacterium. So what is happening over here? What is happening over here? Transformation is happening over here, right? A new thing is being formed. It is amplification, cloning, transformation, or restriction. What do you think is the answer for this? It is going to be C. It is going to be C. Ashwin says it is C. Yes, Baba, it is C, transformation. Okay. It's very, very, very important. Okay, it's very, very, very important. You want red? Okay, I'll make red. Okay, perfect. Let's go. Autonomously replicating circular extra chromosomal DNA is called autonomously. That means it is replicating by itself only. Okay, by itself only. So autonomously replicating circular extra chromosomal structure is known as what? Vector, restriction enzyme, marker, or plasmid. We just studied extra chromosomal, autonomously independent replication. What is it, my dear children? Third answer is D. Rizwana says third is D. What about others? What about others? Spandana says D. Okay, okay, okay. Itzaiba says D. Okay. Purvi, Aksa, everyone is saying D. D is the answer. It is what? It is definitely going to be plasmid. Fine. Perfect. Let's go ahead with the next question out there. Plant cells are bombarded with high velocity microparticles of gold and tungsten coated with recombinant DNA in a method that is known as. Remember, over here you have gold and tungsten filaments. You are wrapping up with the DNA out there. What do you call this method when you are finally going to do this? Is it known as microinjection, biolistic, heat shock, or vector mediated? What do you call this as? Poll is not there, sadly. Soon we will try to inculcate a very important software for that. Fourth B is the answer. You will always be our favorite teacher. Thank you so much. Fourth B is the answer. Oh, so here they have messed up the option. It is B. So B is what? Biolistics. Biolistic is the same thing that is known as gene gun also. So this is your answer. It is used for transferring things into the plants because plants have cell wall and you want to get through that, right? Yes. The commonly used vector for cloning genes in animals is, if at all you're talking about a vector and you're talking about animals, what is it? Rhinoviruses, disarmed retroviruses, disarmed TI plasmid, or Agrobacterium tumefaciens. What do you think is the answer for this? What do you think is the answer for this? Commonly used vector for cloning genes in animals is? Okay, Vijay. Hello, Shreya. Okay, so here's different names. 5B. 5B is what? Okay, 5B, Spandana, Sana. Okay. 5B is disarmed retrovirus. Exactly, it is disarmed retrovirus. Retrovirus, you know, HIV is a retrovirus. What does it do? It transfers its genetic material into our body. The same logic we want. We want to transfer the gene of interest. So we are going to use retrovirus and it is definitely going to be disarmed also. Perfect. Okay, now next one is enzymes which remove nucleotides from the ends of the DNA are enzymes which are going to remove nucleotides from the ends of the DNA but not in the middle. Okay, endonucleases, exonucleases, DNA ligase, DNA polymerase. What do you think is the answer for this question? What do you think is the answer for this question? Sanjana, Sanju, yes, I am. I'm seeing you, okay? Spandana, Rizwana, okay? Rizwana says it is B. Sanjeevani says it is B. Habiba says, okay. Zahiba, sorry. And Indra, okay, everyone says it is B. B is what? Exonuclease. Exonuclease cuts it from the end, so obviously it is going to be correct one. Restriction site for EcoR1 enzyme is, guys, we have to be a little faster. Okay, restriction site for EcoR1 enzyme is what? 5-GAA-TTC 3-3-3-A-TTC 5-. What do you think is the answer over here? Please look for the polarity as well. Dilip Kumar, I said your name. Yes. Yes, and B also, right? Perfect. 7th question, my dear children. 7th question, anyone have given the answer? A. Rizwana says 7th answer is A. Okay. What about others? Haiba says A, and okay, Rahul says D. Okay, Rizwana, Prem, Raj is there. Hello, Prem. Spandana, everyone is saying A. A is mostly what? 5-GAA-TTC. Is that correct? Yes. Ga, T, C, and it is from 5- to 3- direction. 6 base pairs are being involved over here. This is a restriction site for EcoR1. Where will it make the cut? Between G and A. Between G and A. Always remember that. Now, the technique used to separate DNA fragments in recombinant DNA technology is, if at all you want to separate the DNA fragments, what do you use? Gel electrophoresis, PCR, autoradiography, or downstream processing? Gel electrophoresis, PCR, autoradiography, or downstream processing? What do you think is the answer for that? Yes, Sanjana, Sanju, Sanjana, Prem, Raj, Yash. Ma'am, I'm scared. Why to fear da? Being scared, okay, I agree. You should acknowledge that emotion, but you should not drown within it, okay? You should go ahead. Okay. 8th A is the answer. 8th A is gel electrophoresis. Exactly. With the help of gel electrophoresis, you can separate the fragments. The fragments that are going to move farther and faster is the smaller fragments. If at all you are asked which one moves first, you do not know. It can be large, it can be what do you call small also, but the one that moves farther is basically small. Okay, which of the following is the stain used to visualize DNA fragments in gel electrophoresis? You cannot see the DNA fragments, Chumma. You need to visualize with the help of a enzyme, sorry, not enzyme, a stain out there. What do you think is the answer for that? Sanjini, Sanjana, Spandana, everyone, you guys are doing absolutely great. Prem, Shweta, everyone. Mansi, Salome, Jessie is there. Hello, dear. Okay. Mansi says 9th B is the answer. Spandana says 9th B is the answer. 9th B is what? Ethidium bromide. Exactly. Ethidium bromide gets into our DNA, and that's how it gives a bright orangish red color. It gives a bright orangish red color. Your answer for this question is definitely going to be B. Perfect. Now we go ahead with the next question out there. 10th question. The extraction of separated bands of DNA by cutting out them from the agarose gel is. From the agarose gel, if at all you're extracting the DNA out. Okay, what do you call this process of extraction of DNA out? Any idea? Any idea, guys? Denaturation, fragmentation, elution, separation. What do you think is the answer for this particular question? Ma'am, feeling anxiety for HOTS and MCQs. Listen, and that's what I said. Being anxious might not help as of now. You'll be just worried. You might do one of the things, so do not be worried. Stay positive. Try the best to stay positive. 10th C, and I'm seeing so many answers. Srinidhi, Uma Devi, hello, it's Zaiba, Rizwana, Spandana, Sanjeevni, oh my god, Sanju, and all of it. Listen to me, the answer for this question is elution. Elution is the process by which you're going to remove the DNA out of the agarose gel. Okay, the thermostable DNA polymerase enzyme is obtained from. Thermostable means it can sustain high temperature out there. Where do you get that from? Agrobacterium tumefaciens, Thermus aquaticus, Escherichia coli, Melodigon, Incognitia. What do you think is the answer for this particular question? Srinidhi, perfect. Joswin, hello. Okay. MD Mujib, everyone. New names are there. Perfect. Guys, what do you think is the answer? 11B. Sana says 11B. Rizwana says B. Okay. Mansi says B. B is what? B is known as Thermus aquaticus. Exactly. T from here, AQ from here. That's how you get the TAC polymerase, which is thermostable in nature. This is the answer for the question. 12th question. In the naming of restriction enzyme, the first letter of the name is derived from. First letter, for example, when you talk about EcoR1, the first letter over here is derived from what? Escherichia. Escherichia is what? Genus name, right? So species, genus, strain, or family. What do you think is the answer for that? I have already told the answer, but still think about it. It is going to be genus name. Okay, so 12th answer is going to be B. That's an easy question. So, but yes. And the second two letters are from your species, and this is basically the strain, and this is basically number of discovery. Perfect, guys. Okay, so the next question that we have is 13th question. Restriction enzymes belong to the class of. Restriction enzymes belong to the class of what? Oxidoreductase, ligases, isomerases, nucleases. What do you think is the answer for this question? Restriction enzymes belongs to which class, Baba? Hello, Salmani. Okay. D. 13th question, 13D. Sanjeevani says 13D is the answer. It's Zaiba, Prem, Spandana. Everyone says D is what? Nucleases. Exactly. They are going to cut the nucleic acid, which is why you call them nucleases. Endonucleases, exonucleases, right? The answer is D. Now, 14th question. Agrobacterium tumefaciens transforms normal cell to tumor by delivering a piece of DNA. So, agrobacterium says like, Macha, you have this. You are going to get tumor. What is that thing that is being passed over here? What do you call that? 14th question. ZDNA, CDNA, tDNA, rDNA. Let me see who wins Crorepati. Ma'am, I need help. Tell me, Rohit. 13th D. Okay, 13th D. Yes, you guys are correct. Josephine, Sanjana, everyone, Prem Raj. So the thing is that your chat comes, I mean, what I say is a bit, late. What I say will be coming a bit late to you guys, which is why I would have to wait for some time. Okay, Sanjeevani, Jia, Yash, Zaiba, Mansi, Suresh, Spandana, everyone. Okay, hello, hello, hello. So the answer is tDNA. It transfers the transfer DNA. tDNA stands for transfer DNA. It is a part of tumor-inducing plasmid. So your answer for this question is going to be C. Perfect. Now, recombinant DNA is directly injected into the nucleus of an animal cell by recombinant DNA, which is there. It is directly injected into the nucleus of an animal cell by which method? Microinjection, gene gun, insertion, biolistics. What do you think is the answer for this particular question, Macha? Fifteenth A, Selvin Anand. Selvi Anand says 15A, okay. Zaiba says 15A, okay. Premraj, Rizwana, Sanjeevani, Sanjana, Nikesh, okay. So answer for this question is actually A. Micro injection. Recombinant DNA can be inserted directly into the animal cell. Why? Because, Macha, listen to me, animal cell does not have a cell wall, which is why you do not need a gun, and basically gene gun is also known as biologistic. So you don't need all this thing, you just need A. Done. That's all. Now, 16th question. Agrobacterium tumefaciens is used as a vector for cloning genes in. For cloning genes in what? Plants, animals, bacteria, viruses. Agrobacterium tumefaciens, my dear children, it's a virus. It's a bacteria. It's a bacteria, and you need this as a vector in which? Plants, animals, bacteria, or the viruses. What do you think is the answer for this? Ma'am, yes, Gia, tell me. Ma'am, for scoring 65 and tomorrow, will you please come live with all expected and guarantee questions, which would be really helpful. Listen to me carefully, dear. If I even try for it, it might not be possible to do so with this limited amount of time. But I'll still recommend you to join the WhatsApp channel. There I have given you a video that you have to watch. The video is pretty long. I don't think now we have time for that, but still try for it. At least go through the PDF of it. Join the WhatsApp channel first. You will see it there. You see a whole big message. I have written that. Ma'am, Kit Kat break. Ayo, dear, if it's okay, is it okay if at all I do not give you Kit Kat break as of now? The reason is that basically I have to go. Okay, 9:30 I have told, probably it might go up to 10. Okay, not beyond that. There are so many people out there, as in security uncles and so many people, they also have a lot of things, yeah. So, yes, okay. I'm so sorry for that. Yes, I hope you understand. Okay, so you guys were giving me the answer for this particular question, and that is 16th question. It is the one that Agrobacterium tumefaciens is used as a vector for cloning genes in what? Plants. It is the one that is used for plants. Okay? Okay? 16th A is actually the answer. Now, the next question, 17th question. Any protein encoding gene is expressed in a heterologous host is called as. You try this. This is from your NCERT directly. This is directly from your NCERT. Any protein encoding gene is expressed in a heterologous host is called as recombinant DNA, recombinant RNA, recombinant protein, recombinant polysaccharide. What do you call? What do you mean by heterologous host? As in any other host. For example, I am taking from this bacteria, I am putting in some other bacteria. So the other bacteria is the heterologous host out there. So any protein encoding gene, when I am putting it, a gene that is coding for protein, when I am putting it in another host, what do you call this as? People saying model paper are not enough tomorrow's session. What's your opinion? Even I am not sure about model question paper coming out there, most of things. But yes, concepts could be repeating. So make sure you practice as much as possible. Okay. Now, guys, Zephyr is please don't spam. Okay, guys, children, answer for this question. 17C, yes, says 17C, okay. Rizwana says C, Sanjana says 17B, okay. Different answer. Why B and C? B and C is what? Recombinant RNA and recombinant protein. The answer for this question is going to be recombinant protein. Arey Baba, you are putting a protein encoding gene, right? So it will be known as recombinant protein. This is directly from your NCERT. Okay, this is directly from your NCERT. Very important. Okay, cool. Don't write 17B. 17B is not the correct answer. How can recombinant RNA be the answer? It is recombinant protein. Cool? Yes. Rishikesh, yes, dear. Correct. Now next, HIND2 cuts DNA molecule at a specific point by recognizing a specific sequence of. HIND2 was the first restriction enzyme and it is going to explain 14th again. 14th. Okay, I'll come to that. Okay. Once let me do this, I'll come to that. Jia. Okay, HIND2 cuts DNA molecule at a specific point by recognizing a specific sequence of how many base pairs? How many base pairs? Fatima says 6. Okay, Fatima says 6 base pairs, which means your answer for this question is going to be B, right? It is going to be B, exactly. It is 6 base pairs. HIND2 has a restriction site that cuts the DNA having 6 base pairs. So that is absolutely correct. B is your answer for this particular question. Rizwana, all of you, correct. Okay, now 19th question which is out there. Which of the following is the correct order of processing of PCR? Annealing, denaturation, extension, denaturation, extension, annealing, denaturation, annealing, extension, and then what do you have? Extension, annealing, denaturation. What do you think is the answer for this particular question? You have to tell the correct order of processing of PCR. 19th C, Selvi Anand says 19C, okay. Sanjeevani says 19C. Sana Rahat says C. Spandana says B, okay. Sanjeevani says C, okay. Nikesh says C, Hassan says C. Okay, guys, so over here, C is what? Denaturation, annealing, and extension. Exactly. So instead of, okay, I usually remember DNA, right? We have DNA. So over here, annealing has 2 N. So always remember, after denaturation, you have annealing, and after that, you have extension. Does not make any sense? Yes. Okay. Done? Done. Next, going ahead with the 20th question out there. In the given diagram of PBR322 vector, identify the restriction sites A, B, C, and D. What is A? A is the easiest one. If at all you are able to understand A, you will be able to do any of the questions out there. Okay, Mansi, every one of you correct. Julie also is there. Circuit, no. Oh my God. Different name. Nice. Okay. Manjunath also see. Ma'am, you're beautiful. Thank you so much, dear. You know, beauty is in the eye of beholder. I always say that. Still sucking onto that. Okay. Guys, please give me the answer as number with the number. 20C. Sanjeevani says 20C. Okay. What about others? Sanjeevani says 20C. Snell Plays is there. Hello. Okay. Selvi says 20C, oh 20B, okay. Rizwana says 20B. B is what? B is this. So listen, A is going to be origin of replication. This site is basically ROP site. This is tetracycline resistant site. Tetracycline resistant. This is what? Ampicillin resistant site. Over here you have 2 Dundee like this. Always remember this is going to be CLAV1 and this is going to be HIND3. So you have been asked about B. After A is ORI, B is ampicillin resistant, C is going to be tetracycline, and last one D is going to be what? HIND3. You just have one option with A as ORI. So if at all you are able to identify this, you will anyway will be able to get the answer correct. So B is the answer for this particular question. Ma'am, this is the last live, ma'am. Yes, for today, this is going to be definitely the last live. Otherwise, probably this would be the last live for me. Okay? Yes. Super tired, children. Super tired. 21st question. Statement number 1 says, sticky ends are resulted during the action of restriction enzyme. Statement number 2, stickiness facilitates the action of DNA polymerase. What do you think is the answer for this particular question? Come on. Suchitra, ma'am, I'm unable to understand. If at all you have any doubt, you can ask. If at all it is concept related, I'll request you not to stay over here, because I am literally saying you might feel tensed if your concepts are not really good. Okay, so make sure you have your perfect revision first, and then we can go ahead with this. Okay, We will miss you, ma'am. Oh, that's so sweet of you. Thank you. Okay, 21A. Spandana says 21A. 21C is said by Rizwana. Okay. Ma'am, will you do fill-ups also? No, no fill-ups would not, fill in the blanks will not be done over here, dear. Okay. So 21, the answer is, listen, sticky ends are resulted due to action of restriction enzymes. Correct. Okay, restriction enzymes cut them. That's why you got the sticky ends. Stickiness facilitates the action of DNA polymerase. Wrong. Stickiness facilitates the action of DNA ligase. Ligase is the one that joins, right? So it helps the DNA ligase, not DNA polymerase. This statement is going to be wrong. So can I say statement 1 is true, but statement 2 is false, which is why your answer for this question is going to be A. It is going to be A. 7th, 21st answer is A. Kavana, yes, you are correct, dear. Shweta, you are also correct. Selvi, everyone who gave me the correct answer for the 21st question, I hope you have understood it. And those who got it wrong, I hope you have understood as well. Hi, what is your motivation to do this for us? My motivation is that you become happy at the end. So that's all. Yes. Now, 22nd question. I think few more questions are there. Yes, finally. Okay, let's go. The purpose of stir tank bioreactor is, the purpose of stir tank bioreactor is what? Providing anaerobic conditions in the culture vessel. B, purification of the product. C, addition and proper mixing of the preservatives. D, availability of oxygen throughout the process. What is the purpose of stir tank bioreactor? Remember, you have a stirrer to stir things. But why do you need to stir things? What is that? Ma'am, attending your for the last time. Tomorrow morning is there, da. If it all is there, you can come. Okay. Ma'am, I doubt whether it's necessary to write the numbers present alongside the pyramid in the exam. Yes, 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 you have to. If at all that is being asked, yes. Okay. 22B, 22B and 22D. Okay, Shweta says B and Dilip says D. Most of us are saying D. D is what? D is availability of oxygen throughout the process. Exactly. The purpose of stir tank bioreactor is to make sure that the oxygen is available. That is why basically you have flat bed impellers, and that is a part of the stirrer. Stirrer makes sure that availability of oxygen is there. 23rd question. Identify the correct statement. Hind 2 cuts the DNA to specific sequence of 6 base pairs. We just studied it. I am solving this for you. Please make sure you listen to me. Okay. Next is microinjection is the technique of introduction of rDNA into the plant cell. Is that so? Is that so? Okay. No, Baba. Plant cell, are you talking about microinjection? No, it is not plant cell. It is going to be animal cell. Now, biolistic is the technique of introduction of rDNA into the animal cell. Ta-da-ta-ta. Wrong statement. Biolistic or gene gun is the process of introducing to the plant cell, right? Now, retroviruses are commonly called cloning vectors in the plants. Retroviruses are commonly called as used as cloning vectors in the plants. No, they are therefore animals. So your only correct answer for this particular statement is what? A. 33. So 23A. Yes, Rupa. Yes, Vinut Nayak, Spandana, and Shavar, everyone. Dilip, Rahul Raj, Sanjeevani, Shashi, Lishita. Next go. No, CS will not be there, dear, tomorrow morning. Okay. My live will be there at 5:30 a.m. Okay, I'll try to be corrected by that particular time, but I'm not sure in the morning how is the transportation going to be, but yes. 24th question, statement number 1, plasmid DNA act as vector to transfer the piece of DNA attached to it. Statement number 2, vector DNA act as plasmid to transfer the piece of DNA attached to it. What do you think is the correct answer for this particular question? 24th. Come on, guys. Come on, let's go. What do you think? 24th A. Rizwana says the answer for this is what do you call A? Dilip says A. 24. Swandana also says A. A is what? A is basically statement 1 is true, statement 2 is false. Let's see. Plasmid DNA acts as vector to transfer the piece of DNA. Exactly. Plasmid is like this. Here you can put the gene of interest and it will be acting like a vector to transfer. Correct. Statement number 2. Vector DNA act as plasmid. No, listen. Vector can be anything. It can be retrovirus. It can be anything. No, it is not necessarily going to be plasmid always. Not all vectors are plasmid. Remember that. So vector DNA act as plasmid. Wrong statement. So you have statement 1 which is correct and statement 2 is false. Your answer is going to be A. So those of you who gave A as the correct answer, perfect. Okay, 25th last question of this chapter. A single strand of nucleic acid tagged with a radioactive molecule is called. If at all I have a nucleic acid and it's tagged by some radioactive stuff, what do you call this as? Vector, marker, plasmid, probe. Vector, marker, plasmid, probe. Anik, yes dear, you have to draw the Lacopron diagram as well. And Miss change the chapter, will definitely be doing that. 24D, Spandana says, Sanjeevani says, Mansi says, okay, different answer from Mansi, okay, Dilip Kumar also has a different answer. So there's a confusion between B and D, is it? So the answer for this question is going to be probe. It is basically probe. The single strand of nucleic acid tagged with a radioactive substance is basically known as a probe. Perfect. Everyone, give a tap on your back. By the way, it's 9:30. Okay. It's a probe, okay? Just give me a second. I need to make an urgent call out there. Guys, I hope I'm audible now. Come on, 2 more chapters and then we are done. Biodiversity and conservation again gives you some questions out there, two questions. So the term biodiversity was popularized by, who popularized the term biodiversity? Guys, this chapter holds easy questions. Sometimes for some people it feels difficult also. Ma'am, you're looking tired. Definitely I am. No doubt for that. Ma'am, Kit Kat break. 1 more chapter and then we are done with the live altogether. All the 3 and 2 marker questions, 2 MCQ question chapters have been done. Okay, ma'am, you're back, yes. A, please give the answer like 1 A, B, C, D. A is what? Edward Wilson, exactly. He was the one who popularized the term biodiversity. Second question, Robert May places global species diversity at above. Robert May, my dear children, he said that global species diversity is about 2 million, 1.5 million, 2.5 million, 7 billion. What do you think is the answer for this question? Come on. Come on. What do you think is the answer for this particular question? 1A, okay. Sanjana and Kavana, hello. 2, guys, please answer in A, B, C, D, sorry, 1, 2, 3 format. It will be easy for me to understand. Spandana says 2D, okay. Hassan says 2D, Vinayak says 2D, Kavana says D, and someone has different answers, B and all. Manjunath, okay, listen to me very carefully. The answer for this question is going to be 7 million. Robert May, 7 million. Robert May, 7 million. Please remember, there is no other way of helping with it, okay. Global species diversity is 7 million. Now, as per species inventories, 70% of the species recorded are. So basically, when you talk about all the species together, 70% of them, my dear children, the highest of them are going to be what? Plants, fungi, bryophytes or animals. Bryophytes or animals. What do you think is the answer for this question? 2D, Yuvanath and Mansi and Sanjana, Rahul, Ravi, Prithvi, Jaggu and Rohan, everyone perfect, okay? We're talking for the next question. What about the answer for that? 3D. 3D, Spandana, Prithvi, okay. Okay, D is what, Baba? D is animals, exactly. 70%, as in most of the organisms which are there, they are going to be animals. This is going to be the correct answer. Now, the most species-rich taxonomic group among the invertebrates. Now, in animals, you have vertebrates and invertebrates, right? Now, when you're talking about invertebrates, which one are going to be the one with highest number? Any idea? Any idea? 3D, Rasya, as well as Sanjana, Nikesh, perfect. Fourth question, guys. The most species-rich taxonomic group among the invertebrates is what? Fourth B, Rahasya says fourth B is the answer. Okay, what about others? Fourth D, Soundarya says fourth D. Okay. Vinayak says fourth, insect. Exactly, it is going to be insects. Remember, insects are the ones that are found in the most amount in the case of animals, whether it be invertebrates or vertebrates. It is going to be insects, but they come under invertebrates, do not have the vertebral column. Okay, next set of questions that we have. The most species-rich taxonomic group among the vertebrates is, when I talk about the vertebrates, which is the one that has the most species? Algae, fungi, angiosperms, birds, insects, mammals, or fishes. The most species-rich taxonomic group among the vertebrates is what? Birds, insects, mammals, or fishes? What do you think is the answer for this question? Any idea? Fifth D. Okay, hello gaming. Fifth A. Sanjeevani says fifth A. Sanjana says fifth D. Okay, different answers. What is D? What is D? It is going to be fishes, it says. Is it? Yes. So listen, always listen. Farm. Okay, fishes, amphibians, reptiles, mammals. In between you have birds also. So fishes is the one that has the most amount in the case of vertebrates. In the case of vertebrates, they are all vertebrates, and basically in that you have fishes. Okay, sixth question. The most species-rich taxonomic group among the plants are. Now among the plants, which is the one which has most of the species out there? Any idea? What do you think is the answer for this particular question? Senorita Rohan, oh my god, what's happening there, guys? Come on, Macha. Most species rich one. Sixth question, what do you think is the answer for the sixth question? Guys, be on the sixth question itself. Sanjini says the answer for sixth is B. Sixth is B. B is what? Fungi. B is going to be fungi. Okay. B is going to be fungi. That is the answer, is it? Yes. So in your NCERT, they have mentioned fungi in plants only, and they are saying that fungi is the one that is in highest amount in the case of plants. And always remember, fungi is actually the number of fungi which is there in our earth is the total of all the fishes, amphibians, reptiles, and mammals put together. Farm put together. Remember that also. Now, the share of global species diversity of India. Global species diversity of India is what? 8.1, 8.5, 9%, 2.4%. What do you think is the answer for this particular question? What do you think is the answer for this particular question? Seventh A. Okay, seventh A, Vinayak, Snell, and as well as Sanjana. Okay. Seventh A is what? 8.1%. Is that so? Yes. So India's diversity is basically, India contributes 8.1%. India as a whole only have 2.4, occupies 2.4%. But you know what? It gives 8.1% species diversity, and that is commendable. Not 8.5, it is 8.1. 8.1. Okay. Yes, the species area relationship was given by Edward Wilson, Robert May, Alexander von Humboldt, or Paul Ehrlich. Who was the person who gave the species area relationship? A, B, C, D. 8th C. Chinnappa says 8th C. Is it? 8th C. Chinnu also there. Chinnu, hello Chinnu. My mother's name is also Chinnu. Okay, Chinnappa says C. Okay, guys. What about others? Soundarya, C. Dilip will be having later on. Thank you for asking. Kumada says C. Chinnappa says C. Okay, Gabing also sees Jaggu, Yuvanath, Blashi, Sanjeevani. Everyone says C. C is Alexander von Humboldt. Yes, he was the person who gave species area relationship. S is equals to CA raised to the power Z. Perfect. Now over here, in the given species area equation, log S is equals to log C plus Z log A. The letter Z represents what? What does Z represent over here? Area, species richness, regression coefficient, or the Y-intercept? What do you think is the answer for this particular question? By the way, everyone is doing absolutely great. Perfect. RMS, hello. 9th D, 8th C, okay. 9th C, 9th D, gaming says D, okay. So 9th D is something that most of you are saying. Is it D? No, it is not. It's not the Y-intercept. Z represents regression coefficient. Z represents regression coefficient, my dear children. If at all you have attended today's live lecture, you might still definitely know it. Sanjeevani, Sanjana, Alizaga, So guys, you tell me, the live lectures were useful for you, right? Some of you might be feeling that while you're giving the answers. If yes, let me know here itself. Okay, so it is regression coefficient that is there. Always remember that. Z represents that. Okay, now 10th question. According to ecologists, the value of Z lies in the range of. So the usual value of Z lies in which range, my dear children? The usual value of Z, 0.1 to 0.5, 0.2 to 0.6, 0.6 to 1.2, 0.1 to 0.2. What do you think is the answer for you? Hi ma'am, great salute to you. That's so sweet of you, dear. Very useful, ma'am. Thank you so much. A lot. It was very helpful. Thank you so much, Jia and Vinayak and everyone. Shower, everyone. So 10th D is what you are saying. 10th D is what? 0.1 to 0.2. Yes, the answer is that. Always remember 0.1 to just 0.2. Only 0.1 gap is there, and this is the usual range which is there. But in some of them, the area when it is area is large, my dear children. In this particular case, the species area relationship, the Z value is going to increase. What is the increased value? It is 0.1 to 0.5, 0.2 to 0.6, 0.6 to 1.2, or 0.1 to 0.2. This is the one for smaller areas usually. So you have been asked about larger areas. What do you think is the answer for 11th question? 11th question. Guys, all of you are giving me 10th question answer. Perfect. Rizwana, Mansi, Jia, Sanjeevani, all of you. Perfect. Jaggu. Okay. Chinnappa says C is the answer for this question. Okay. Spandana says C. C is what? Maruti Mane is also there. Says C. Yes, it is going to be 0.6 to 1.2. I'll tell you a way to remember it. 6 plus 6 is 12, right? So over here, instead of 12, you just have a 0.2. Okay, point given 0.6 to 1.2. Now listen to me. Slope of line regression coefficient for fruit eating birds and mammals in the tropical forest of different continents is found in the range of. So for fruit eating mammals also, the Z value is usually high. That means it will be coming in this particular range only. It will be coming in this particular range only. Now you tell me the answer. What will be the answer? A, B, C, D. What do you think is the answer for fruit eating birds? Listen to me. Usually it is just 0.1 to 0.2. In the air, where the area is more, it will be 0.6 to 1.2. And now when you're talking about the fruit eating birds and stuff like that, obviously it is going to be the same only, the larger one only. 12B, exactly, Rizwana, exactly, Sanjeevani, Spandana, Sanjana, Mansi, everyone. Perfect. Chinnappa. So the answer for this question is going to be 0.6 to 1.2. If at all you are asked about specific value, it is actually 1.15. 1.15, and that comes under 1.2, right? 1.15 is the actual one over here. Always remember that also if possible. Cool. Let's go ahead with the next set of questions that we have. 13th question. The rivet popper hypothesis was proposed by Paul Ehrlich, Alexander von Humboldt, Edward Wilson, and MacArthur. Who do you think? What is the thing? What is the answer that you think is correct? Who gave the rivet popper hypothesis? He said that ecosystem, or basically everything that is over here, is like a what do you call, aeroplane. Yes, and if at all you remove those rivets out there, it is going to be not sustaining. Hassan, ma'am, you got to rest now, definitely. Okay, so what do you think is the answer for this question? 13th A is the answer, right? Paul Ehrlich is the answer for this particular question. All of you gave me the correct answer. Kudos. Next, which of the following species introduced for aquaculture purposes is posing serious threats to the indigenous catfishes in our country? Indigenous means they were already there. You got something from outside. For what? For aquaculture. But now it is posing like a threat. What is it? Clarius, Garipinus, Ecrinia, or Parthenium, or Lentana? Listen, my dear, I told you I brought this for you, right? This is what? This is Parthenium. This is plant. This is not catfish. Similarly, Lentana is also basically plant, and here it should have been Ecornia. I don't know why it is not like this. So anyways, the only thing that you're left with is what? 14th A, exactly, Hassan and Spandana and everyone out there. Kudos. Sanjana, Sanjeevani, Isaac, Alizak. Okay, everyone, Prajwal, perfect. Rizwana, Rahasi, everyone. Clarius Garipinus is the name for it, and it is also known as African catfish. Okay, Yes, the narrowly utilitarian arguments for conserving biodiversity are, according to narrowly utilitarian point of view, why should we conserve the nature? Narrowly utilitarian means they're thinking like this only. Why should we conserve the nature? Because they will get direct benefits. So you have to think about the products, okay? Whenever we are conserving the nature for some products of the nature, that is known as narrowly utilitarian point of view. So cereals and fruits, ecosystem services, intrinsic value, aesthetic pleasure. Which one do you think is coming under product? You give me the answer for this. 15th is Sanjana says 15th A, Spandana says 15th A, Sanjeevani 15th A, Rizwana and everyone out there. Blushy Salim. Okay, so your answer for this question is going to be cereals and fruits. Cereals and fruits. That is why we should be conserving nature according to narrowly utilitarian point of view. If it was broadly utilitarian point of view, then we should be conserving it for ecological services. For example, atmosphere, air, aesthetic value and stuff like that. Okay, if intrinsic value, that means ethical reason. Cool. Now, the 16th question out there. The following are the in situ conservation approaches except. The following are the in situ conservation approaches except national parks, wildlife sanctuaries, biosphere reserves and botanical gardens. What do you think is the answer for this? Prerana is there. 16th D is the answer. 16th D is it? Botanical gardens. The following are in situ conservation approach except botanical gardens. Exactly. So botanical gardens, we take the plant from elsewhere and put it elsewhere, right? Everything else is being conserved in their natural habitat. So this one is the actual correct one. Awesome. Okay, ma'am, I'm Yasmin. Okay, sorry, Blushy, I'll try to remember that name. The following are the ex situ conservation approaches except. The following are ex situ conservation method except you have been asked, right? Zoological parks, seed banks, sacred groves or botanical gardens. So which one is not the one? Which is not the one? Ma'am, should I solve model question papers? Yes, definitely. 17C, Hassan says 17C, Mansi says it is A. Okay, gaming says it is C. Okay, Bharat says it is B. Okay, you're giving me options. Okay, the answer for this question is sacred groves. What do you mean by sacred groves? For example, today I have a place over here and I feel it is religiously very important to me. If I'm safeguarding that place, obviously all the organisms up there are also being safeguarded. It is conserving the organism in their natural habitat. Okay, in their natural habitat. So over here your answer for this question is going to be C, sacred groves, because this is an in situ conservation method. Now Chanda and Bastar areas known for sacred groves are found in Chanda and Bastar, my dear children, both of them are sacred groves and they are known to be found in what? Meghalaya, Madhya Pradesh, Karnataka, Rajasthan. What do you think is the answer for this question? What do you think is the answer for this question? 18B. Selvi says 18B. Okay, Sanjana also says 18B. Sana Rahat also says 18B. Hassan says 18B. Prerna says B. Okay. MP, that is Madhya Pradesh. Yes. Chanda, Bastar, Sarguja. Chanda, Bastar, Sarguja are from Madhya Pradesh itself. They are all sacred groves of Madhya Pradesh. Perfect. Now, the Earth Summit was held in Rio de Janeiro in. Earth Summit, my dear children, was held in Rio de Janeiro in which year? 1991, 1992, 210 or not 2010 and also 2002. What do you think is the answer for this? Hi ma'am, good night shift ma'am. Yes. I recall that you lived in Indore. Oh, that's so sweet of you, Hassan. Yes, exactly. Ma'am, you look literally tired. You need sleep. Definitely. We all need sleep, don't we? Okay. 19th A, Sanjana says, Mubarak says, okay, Pradeep, Maruti, Prerna, Rahasya, okay. Answer is going to be 1992. Okay, always remember 1992, Earth Summit happened. You just add 1 over here, it becomes 2002 and that's where World Summit happened. 2002 World Summit, Earth Summit 1992 in Rio de Janeiro. The World Summit on Sustainable Development was held in 2002 in what? Canada, Johannesburg, Brazil or Tokyo? Canada, Johannesburg, Brazil or Tokyo. What do you think is the answer for this particular question? What do you think is the answer for this particular question? Any idea? Kavana says 19B. Okay. Hassan says 20th B. 20th B was Johannesburg. Exactly. It is going to be Johannesburg. That's where the World Summit happened. The Earth Summit happened in Rio de Janeiro. Perfect. Okay. Yes, yes. Going ahead with the next question out there. Statement number 1. There are 32 biodiversity hotspots in the world. Statement number 2. High level of species richness and high degree of endemism are criteria for declaring a region as biodiversity hotspots. What do you mean by speciesness? That means too many amount of species are there. And high degree of endemism means these organisms are not found elsewhere. They're exclusive species out there. And whenever an area has all these things, that's when we say that basically this is a biodiversity hotspot. So what do you think is the answer for this? Hassan says 34 biosphere, not 32. Is it? Yes. So what do you think is the answer for this? 21st question. 21st question. What do you think is the answer for this particular question? Sanjana says 21st A. B. Bajrangi says 20B. Gaming says 21D. Okay, 21 now. Yeah. 21. 21D is the answer for this particular question. Statement 1 is incorrect, but statement 2 is correct. Why, macha? Because over here 32 is written. It is 34 biodiversity hotspots, not 32. And there are 2 from India. One is Western Ghats, another one is Himalaya. Okay. 22nd question, and with that we finish up this particular chapter also. Statement number 1. Tropical regions have got a long evolutionary time for species diversification as compared to temperate regions. Is it? Tropical regions, do you think they have got more time for species diversification? Obviously. In temperate mediation, frequent glaciation is happening. For example, I am over here, another day snow comes, I go down. So basically, obviously you're not giving time for species diversification. This is correct. Statement number 2. Temperate regions have undergone frequent glaciation. Exactly. In the past, whereas tropical regions have remained relatively undisturbed for so many years. Now they can twist and turn and give it to you. Please read your options carefully. So what do you think is the answer for this question? Both statement 1 and statement 2 are correct, right? That will be the answer for this question. So 22nd, the answer is A. Okay. Is this clear, sorted for everyone? Exactly, Kani Studio, Hassan, both of them, Kani and Rizwana, Prerna, Kavana, Bajrangi, Mansi, Sanjana, Sanjeevni, all of you, Adarsh, Jia, exactly, yes, perfect, Vinayak, Police, everyone. Okay guys, now we are entering the last chapter for the day, and that is evolution. With that, we'll end up the live. I hope you have got it. All the 2 and 3 marker, I mean, the chapters that gives 2 or 3 MCQs, I have covered them up. Okay, let's go. So, okay, I hope this is visible. Theory of spontaneous generation was experimentally disapproved by. Spontaneous generation means, for example, over here, okay, over here. Yes, definitely. Okay, when we say spontaneous generation, that means, for example, a person said that there is a sack of things being kept over here, and from that I got a mouse. Oh my God, spontaneously got generated. Is that true? Obviously no. And that is why, who came into the picture, dear? Who came into the picture? 21 says. First answer is A, exactly. It is Louis Pasteur who came up with his experiment saying that this is actually wrong. I'll just zoom it a bit more. I think it's not visible. Yes. Now next thing, the thorns of bougainvillea and tendrils of cucurbita represents what? Homology, convergent evolution, divergent evolution, analogy. And over here, the options are given over here. Please understand. Thorns of bougainvillea and tendrils of cucurbita, both of them, they come from the axillary bud. One forms the thorn, another forms the tendril. So over here, both have come from the same place itself. So obviously, they have diverged to do 2 different things. So it is divergent evolution. And always remember, when you have divergent evolution, you have H also. HD and AC. HD and AC. So homologous organs, homology and divergent evolution, your answer would be 1 and 3. 1 and 3, that means option is going to be B. 2B, perfect Sanjana, perfect Vinayak, and Ashish, Kani Studio, Maruti Mane, everyone. Ma'am, shout out. Thank you so much. Okay, next thing. Third question. Statement number 1. Man has domesticated dogs and has created different breeds by artificial breeding. Is there something like that? Exactly. There is something like that, Ria. Because man has domesticated dogs and has done artificial breeding for bringing out different type of plants. Yes. Vamshi, yes, please tell me. Okay, next thing that we have, statement number 2, intensive breeding of dogs signifies divergent evolution. So does it signify divergent evolution? Listen to me carefully. The dogs were of similar types, they have diverged. Now you have husky, you have this, you have that, you have so many things. Aren't they diverging? Yes, they are. So can I say this particular statement is also true? Yeah, in that particular case, 3A, exactly. Your answer is going to be 3A. Both statement 1 and 2 are going to be correct. Prerna, Shrisha, Mansi. Guys, if at all you're getting things wrong, make sure you listen to me out carefully. Rizwana, Vamsi. Yes, yes, please ask without. Cool? Yes. Next question that is there is question number 4. Statement number 1 says antibiotic or drug resistance is due to evolution by anthropogenic action. What do you mean by anthropogenic action? Basically, it means that human-made, man-made actions. So if at all antibiotic or drug resistance is there, do you think it is because of anthropogenic action, man-made action? If we are eating antibiotic and it's still not working for our body because the organisms have been adapted to that, is it because of us? Obviously. You are the reason. I am the reason. So this is correct. Statement number 2. Many antibiotics or drug-resistant organisms arise due to over-usage of antibiotic. So tomorrow, ma'am, I'm not well. Oh, okay, Hussein, eat this drug, eat this particular medicine. It's okay. You will be fine. Day after tomorrow, I'm not well. Eat this. No, it will be totally fine. We do this, and what are we doing? We were making the organism resistant. Okay. So obviously, this is one very important thing. Okay, Snell plays as well as Sanjana, Maruti, everyone. I hope you guys understanding. Shrex, hello. Rizwana, yeah, the answer for this question is going to be both A and B are correct, or 1 and 2 are going to be correct. Now, if you only do all diagrams and 3, I'm not sure, Light of Dean. How can I talk about model paper? There is no clarity whether what is going to happen tomorrow, but yes, keep going. With whatever resources you have, study properly, that's all. Charles Darwin in Galapagos Island found that finches had different forms of beaks. This is the process of evolution. Represent what? When we say that finches which were there, they having different forms of beaks. Natural selection, adaptive radiation, Darwin fitness, struggle of existence. What do you think is the answer for this? So there were beaks initially. Some went there, some went here, some went here, some went here. So what are they doing? They're radiating, Baba. They're radiating, and what are they doing? They're doing adaptive radiation. They are going to do adaptive radiation. Ma'am, will we get 12 marks? 10 to 12 marks, probably. Okay, 5B is the answer. Kani Studio, Usharai and Prerna, Shower, everyone. Exactly. Adaptive radiation is going to be the answer for this question. Next question, 6th question. A brown-billed duck is newly added to a farm of white-billed duck. Listen to me carefully. There were white ones first, and now you're adding the brown ones. What is the thing that is happening over here? Gene flow, genetic drift, mutation, natural selection. What do you think is going to happen over here? Gene flow, genetic drift, mutation, natural selection. Any idea? 6C. Prerna says 6C. Okay. Sanjana says 6C. Okay. What about others? Gaming also say, gaming has a different answer, is it? Okay. And I feel that gaming's answer is actually correct. Gene flow is the one. So brown-billed duck is newly added to a farm. Listen to me carefully. I always give this example. We are Indians. From somewhere else, some other people are coming. We have our allelic frequency out there. We have a different skin tone also. Okay. Now from outside, we are getting organisms, and they are coming over here. Now obviously you're adding up what new genes. These genes are flowing up here. Okay. It's not already here. We are bringing in. This is known as gene flow or gene migration. So your answer for this question is going to be what? It is going to be A. It is going to be A. Okay, very, very, very important thing. 6A, Samya, yes, exactly. Adarsh Gauda, I hope it's corrected now. Mansi, everyone, yes. Change in frequency of genes and alleles is due to. When do you think genes allele frequency is going to change? Any factors that affects genetic equilibrium. Gene flow, genetic drift, mutation, natural selection, genetic recombination, everything. So mutation and gene flow is given over here. So A and B, both A and B option is also there. So what do you think is the answer for that particular question? It is going to be C. Ma'am, zero preparation. Karthik, you didn't do zero preparation. You were studying. Right? I saw you in multiple lives. Okay, 7th C is the answer for this question. Let's go. The first amphibians that lived both on land and water evolved from fishes, turtles, dolphins, and frogs. Listen to me carefully. Farm, fishes, amphibians, reptiles, and mammals. From fishes you got amphibians, from amphibians you got reptiles, from reptiles you got mammals. Now when I say that the first amphibians that lived both on land and water, okay, were evolved from, can I directly say fishes? No. Listen, not all fishes. Some of them, and you have a specific name given in your NCERT, and that is known as lob fin. Why do you call lob fin? Because its fin was this thick such that it could walk with the fin. Okay, it could walk, literally walk with the fin. So over here your answer for this question is going to be lob fins. C is the answer. Exactly, Rizwana and Sanjana, Mansi, Sanjana, Vinayak. Lone fins, lob fins. Okay, yes. Next question that we have is, arrange in correct sequence of their occurrence on geological time scale. Amphibians, fishes, reptiles and mammals. I gave you already. So what do you think is the answer for this? What do you think is the answer for this particular question? Ma'am, will we get diagram in part C tomorrow? 3 marks, yes. From the chapter Sexual Reproduction, Flowering Plants. I hope so. Mansi says 9th C, 9th C. Prerna says 9th C. Okay, C is what? C is this. Okay, C is going to be this. Fishes comes first, then comes amphibians, then come reptiles, then come mammals. Your answer is going to be C. Your answer is going to be C. Perfect, guys. Perfect. Okay, awesome. Now going ahead, what do we have? 10th question. The biggest land reptiles that existed during Mesozoic era was? During the Mesozoic era, in Mesozoic you have Jurassic period, Triassic period and things like that. During that particular time, which was the biggest one to exist? Ichthyosaurus, Tyrannosaurus or sea cows, and also you have crocodiles. Ma'am, I wasted time. Now you still have time. Okay, you have time until the morning. If at all you are someone who have slept enough, go watch the video of complete biology. It might really help you. Okay, 10th B. Gaming says 10th B. Jigar also says B. Hi Jigar. Mansi says B. Kanne Studio says B. Okay, Prerna, Shashi, B. Rahul. Okay, so B is what? Tyrannosaurus Rex. Exactly, this is the one that was the biggest land reptile that existed out there. Next question. Arrange the following in the order of evolution. Neanderthal man, Australopithecus, Ramapithecus, Homo habilis, Homo erectus. What do you think is the answer for this particular question? First thing, human-like organism, Ramapithecus. From them, you got Australopithecus. From them, you got the Habibi, the first human-like feature. From them, you got Homo erectus. From them, you got Neanderthal man and also Homo sapiens. This is a thing that you need to remember. So, Rahen. It is what? Rahen. Rahen. Or whatever name you want to remember. From here, you get Homo sapiens also. Okay, so in that way, what do you think is the answer for this question? 11D. 11D, yeah. 11D is what? C first, okay. Then B, okay. Then D, okay. Then E, yes. And then A, yes. Perfect. Correct. It is going to be 11th D. Awesome. 11th C, matcha. No, matcha, it's wrong. 11th D is the answer. Okay. Koala, Tasmanian wolf, kangaroo, marsupial rat, all of this represent what? Darwin's finches, Australian marsupials, placental mammals, non-placental mammals. What do you think is the answer for this question? 11th question. 11th D. Prithvi, Rahul, Prithvi, Shashi, okay, Sanjay is there, hello. Rohan, everyone says D for the previous question, okay. Come on, guys. 12th B. 12th B is what? Australian marsupials. Exactly. I told you always the bigger name. Tasmanian wolf cat, Tasmanian tiger cat, this whelp, all the things they come under Australian marsupials. Always remember that. Perfect. Next one. 13th question. Statement number 1. Hugo de Vries believed mutation leads to spontaneous evolution. Did he? Yes, he said that. Mutation is there, obviously there will be faster evolution out there. He was a person who vouched for faster evolution. Darwin said everything is going to happen gradually, slow. And second statement, mutations are random and directionless and causes saltation. What do you mean by random and directionless? As in, anything can happen any particular time. And also, my dear children, one very important thing, they are directionless. That means it can be happening for good and bad. That is why directionless. And causes saltation. Saltation basically means one single step large mutation that leads to forming of a new species altogether. That can happen? Yes, definitely. So both these particular statements are true. So can I say, okay, can I say morning 5:30 is what I believe. So over here, the answer for this question is going to be A. Okay, 13th, exactly. Gaming, perfect. Okay, clear children? Are we good? Will I be able to scale? Hopefully, yes. Or you can watch yesterday's and today's live. That would also be more than enough. Okay, yeah, but in that there is MCQs not being covered. Okay, anyways. Elongation of neck in giraffes due to inheritance of acquired character was given by. So one person said that basically there were small giraffe with smaller necks, but they did this, and then finally they had a bigger neck. Okay, and he said that it is happening because organisms who were doing like this, they're acquiring this and then transferring to the next organism out there. But do you think acquired character can be going ahead? For example, tomorrow I go to gym and I have dole shole over here. Do you think my babies will be having dole shole when they are? No, definitely not. Okay, that is not at all possible, right? This is acquired character. This is not inherited. It's not making any changes in my genes, right? So your answer for this question is going to be 14th A. 14th A, exactly. Mansi, Kanni, Nagashri, Sanjana, Vinayak, Mansi, Bhumika, everyone. So the answer for this question is Lamarck. Lamarck was the guy who said that. Okay, does not make any sense? Yes. Okay, the next thing that we have is 15th question. Let me see how many questions are there. Are we going to end it up right now? Yes, we are. 15th question. According to a parent, which one of the following was not present in the primitive atmosphere of the earth? Which one was not present in the primitive atmosphere of the earth? Water vapor, oxygen, methane and hydrogen. Water vapor, oxygen, methane and hydrogen. What do you think is the answer for this particular question? Prashant, Ashtamak, birthday. I cannot say that, dear, because there is no clarity on that as such. But yes, I'm hopeful that most of the concepts will be repeated. Yes. 15th B is the answer. Gaming says, Nagashri also says B, Kanistudi also says B. B is what? Oxygen. Oxygen was not there. It was a reducing environment, okay, in the primitive earth, correct. Single step large mutation leading to speciation is also called as. Single step large mutation leading to speciation is also known as natural selection, branching descent, founder's effect or saltation. What do you call this as? What do you call this as? Come on. Kanni Studio says 15B. Everyone all obviously correct. Sanjana, Navya, every one of you are doing great. Shower. 16th question, guys. Gaming says it is D. Okay, Vinayak says it is D, Navya says it is D. D is what? D is going to be saltation, and we just discussed about it. This is the single step large mutation out there. Yes, that leads to new species formation. Okay, which of the following theories attempts to explain the origin of universe? And there is a series on it. Okay, which one theories is there to explain the origin of universe? Panspermia theory, Big Bang theory, chemical evolution theory, Darwin's theory. What do you think is the answer for this particular question? Yes, guys, you guys are correct when you say D. Dr. Ramina, Rahul, Deepanawale, and Kavana, everyone, Mahadev. Okay, Gaming says the answer is 17th B. Kanistru says it's B. Sana says it is B. B is what, Macha? B is going to be Big Bang theory. Yes, it is Big Bang theory that explains the evolution of, or basically the origin of universe. Next thing, in Miller's experiment, the environment of primitive earth was created and the temperature was very hot. Okay, 400 degrees Celsius, 450, 800 or 850. What do you think is the answer for this particular question? Okay. Second examination, hopefully, yes. There are almost 10-15 questions more. That's all. Okay. 18C. C is what? 800 degrees Celsius. Exactly. In Miller's experiment, the environment of primitive earth was created and the temperature was pretty good. Not pretty good, pretty hot. It was basically 800 degrees Celsius. Very much important. Next thing, the organic compounds found in Miller's experiment was. Organic compounds that was found in Miller's experiment: amino acid, nitrogenous base, pigment and fat. Which one do you think it was found in the Stanley Miller's experiment? Any idea? Okay. 18C, yeah. 19th question, guys, 19th question. 19A, Kani Studio says 19A, Rizwana says 19A, perfect. A is what? Muhammad Javed also says that. Okay. So your answer for this question is going to be A only, that is amino acids. Only amino acids were found, nothing else were there in the Stanley Miller's experiment. Now we go ahead. The paleontological evidence for evolution is obtained from fossils, embryos, homologous organs or analogous organs. What do you think is the answer for this? This. The paleontological evidence for evolution is obtained from, guys, come on, 101 people watching this video. Guys, let me see who can give me the correct answer for this. 20th A. 20th A is the answer exactly. Paleontological evidence means fossils. Any Friends fan over here? Remember? Yes? Very, very important. Okay? Okay. Perfect, guys. Next, fossils are hard remains of life forms found in igneous rocks, metamorphic rock, sedimentary rocks, any type of rocks. Okay, simple question, macha, but very confusing for some of us, because we think when you complex things, we try to complicate the question as much as possible, right? So 21st question, anyone, any idea? Fossils are hard remains of life forms found in what? Igneous. 21C. Gaming says 21C. Karni Studio C. Nagashri says C. Hanumanta also says C. C is what, macha? C is going to be sedimentary rocks. Is it C? Is it going to be C? It is not C actually. Any type of rocks could be there, because in your NCERT they have mentioned any rocks. I mean, they just mentioned rocks, right? They have just mentioned rocks out there. They didn't mention sedimentary rocks as in name. Okay? So it is going to be any type of rock is what I believe, but you can again have a quick check also. I read the NCERT once. They did not mention any. So they mentioned rocks. So that is what I'm saying. Okay? So the fossils can be any life forms that are found in any type of rocks, and yeah. Fine, going ahead with the next question. Statement number 1, the study of fossils in various sedimentary layers indicates the geological period in which they existed. Sedimentary layers are different, yeah? The study of fossils in various sedimentary layers indicates the geological. For example, I found in this particular layer, that means we have come so before, okay? If at all in this layer, that means probably later on these organisms came, right? So this is correct. Statement number 2, ages of the fossil can be determined by radioactive dating. Can it be determined by radioactive dating, Baba? It can be. It can be. There's the only way out there. So can I say both the statements 1 and 2 are correct? Yes. So dinosaurs are deep, we find the fossils deep within, and human beings we find at the top layer. That means that basically these guys came in that particular geological era and we came over here. Okay, so your answer for this question is going to be A. Perfect. The next question that we have over here is what? 23rd question. Embryological evidence for evolution was disapproved by. Embryological evidence basically means what, Baba? Embryological evidence means, yes, ma'am, it's C only, sedimentary rocks. Is it? Would have to check once again, but we'll be checking it before the live ends. Okay, so over here, embryological evidence for evolution was disapproved by Ernst Haeckel, Carl Ernst von Baer, Charles Darwin, and Alfred Wallace. Guys, if at all in that particular case, just keep this on hold, okay? Keep this particular thing on hold. I'll read it once more, but with what I believe, they have not mentioned any rocks there. Okay, so keep this on hold for some time now. Okay? Come on, get unselected, not working. Come on, come on, come on. Okay, and that's a problem. Yes. Works? Yes. So keep that on hold. But yeah, embryological evidence for evolution was disapproved by, guys, any idea? 23B, yes, 23B is what? Carl Ernst von Baer. So Ernst Haeckel was a person who basically gave the embryological evidence, and Carl Ernst von Baer, the one with the bigger name, was the guy who said that this is not actually correct. Okay, if at all you're saying that we have some characters of the fishes, why don't we have some characters of amphibians and reptiles also? In that case, it should have been done like that, right? So 23B is your answer for this particular question. Going ahead with the next question that is out there. Epochs, periods, and eras depicts what? Geological history, biological history, physiological history, and embryological history. What do you think is the answer for this particular question? Mr. and Gaming. Okay, Anand Gaming. Hello, 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 hello. Varun Gada, B. Guys, ma'am, fail exam again repeated, but first exam failed. Can we write the second examination? We'll be making a separate video on that. Yes, yes. Nikhil says B is the answer. Rizwana says 24th A is the answer. What about others? Guys, please give me the answer like 24 and then A, B, C, D. 24A. Now we also say 24A. The answer is actually 24A, geological history. This is the answer for the question. Now, arrange the following plants as appeared on different geological periods. Conifers, Nettles, Dicots and Monocots. Please listen to me. This is very important. There is a graph altogether, not a graph, a diagram which is given in your NCERT. Go through it once about the, about the formation of the plants, how it went, okay? Evolution of the plants. So there you have conifers first and then you have Nettles. Both of them, they are gymnosperms. From them you, both of them are gymnosperms. From them you got the angiosperm, that is dicots, and then you have monocots. Not from them, but yes, they came first and then came these guys. Okay, so conifers, then Nettles, then dicots, then monocots. Both of them are going to be, they are gymnosperms, and these two guys are angiosperms out there. Okay, cool. They are found in Mesozoic era. This is also found in Mesozoic era. This is found in Cenozoic era. Okay. So there is no much to teach over here because yes, I cannot do that right now. I do not have the resources out there, but yes. Okay. Bye. Good night, ma'am. We'll all love you. Thank you so much. Okay. Now, potato is respectively homologous and analogous to sweet potato and tendrils of cucurbita, tendrils of cucurbita and sweet potato, thorns of bougainvillea and tendrils of cucurbita, sweet potato and wings of butterfly. What do you think is the answer for this question? Potato is respectively homologous and analogous to. Homologous means basically they might have diverged from that. Yes, analogous means they have the same function. So over here, potato and sweet potato have the same function of storing it. Yeah, so it is going to be analogous, and it's just a one option where sweet potato is on the second position. So this is going to be the answer. Tendrils of cucurbita, tendrils came from what? From the stem modification. Potato is also a stem modification, right, my dear children? So your answer for this question is going to be B. 26, the answer is B, not A. It is going to be B. Listen, homologous. Homologous means same function should be there, or basically same structure should be there, not function, same structure should be there, right? So over here, tendrils of cucurbita came from the stem modification, axillary bud. Similarly, potato is also stem modification, stem tumor. Analogous means that they will not have the same function. Okay, they will not have the same function. By the way, hello Amma. So my Amma is video calling me. Okay, bye. I have a live. I'll talk to you later. Okay, so that is the answer. Your B is the answer for this particular question. Done? Done. Now, statement number 27. Statement number 1. Pouched mammals of Australia survived because of lack of competition. Is that so? Yes, there was no competition. So there was continental drift that happened. You know, continents, they divided, they got moved here and there. And pouched mammals of Australia, they did not have any competitors out there, which is why they survived. Okay, so continental drift happened, and that is when these guys were left alone with non-competitors. So this is correct. Statement number 2. Continental drift has both positive and negative effect. Yes, it has. Negative effect, I will tell you. So there was basically South America over here and North America. The South American one, because of the continental drift, basically they joined up with North America. What happened? The North American organisms overrid them, and that's how there was a huge amount of competition out there. So continental drift can be useful and also not. Okay, so over here I will say that both statements are correct. Yeah, both statement 1 and 2 are actually correct. 27th answer is A. Exactly, everyone. Perfect. And now we go ahead with the last question out there. Observe the following diagrammatic representation of operation of natural selection on different traits. Here, natural selection depicts what? So over here you have two jumps out there. This means that two extreme characters are being selected. Two extreme characters are being selected. So okay, tall and dwarf, both of them. So what do I say? This is disruptive selection. Stabilization, directional, disruptive, or cannot be depicted. What is your answer for this? It is going to be disruptive. And with that, we complete the live. Okay, so it's been 10:19. Okay. Ma'am, reminding you to hydrate your body, drink some water, definitely doing so. Please make sure, guys, you sleep tight. Okay, sleep early and do not think about too many things. Now what has to happen will be happening. And yes, if at all you are someone who have not prepared yet, you are doing your preparation still, make sure you have something, eat something up, yeah? And please take care of yourself. Everyone who is watching this video, I hope you are subscribed to the channel. If not, subscribe to it right now. Yes, thank you, Warren. I'll be definitely doing that. Navya, do not be scared. Yes. Okay, we love you, ma'am. Thank you so much. Love you too. Take care, and please look up for that Rockwalla question. As what I believe, it is all of the above. Ma'am, you're so pretty. Thank you so much. Bye, guys. We'll be seeing tomorrow morning at 5:30. If not 5:30, then at 6 o'clock probably. We'll be checking through that. We'll be letting you know through the WhatsApp channel. So join the WhatsApp channel right now. Yeah. So yes. Okay, bye-bye. Thank you so much, ma'am, for your hard work and dedication. Mansi, that's sweet, sweet of you. Okay. Take care. Thank you, Rizwanas. Thanks for the efforts. You're most welcome. Bye. Take care. Lots of love. Lots of love. Wishing you the best. Someone was asking, am I married? Come here. Come, no. Come. Patna. What if I pattern Samila? Yes, I'm married and this is my husband. Okay. So, yes, thank you. So, guys, it's time to go. Wishing you the best. Take care. Lots of love.